Hello everyone, good evening and welcome to your very own BYJU'S 9th and 10th grade channel. I'm Aishwarya and today's class is going to be as a part of our Foundation Fest, where we are going to be covering all the chapters that we have studied so far in biology in one single video, right? So we're going to be looking at the chapter Fundamental Unit of Life, we're going to look at tissues and improvement in food resources, yes? So please make sure you have your notebooks, your pens ready with you, you have a bottle of water. I have also put some break time slides in between so that we take a break, we freshen up a little, we take a walk around our room itself, because see, today's class might extend a little long, and once after we quickly cover whatever we've learned so far, we're going to have a quick Menti quiz towards the end as well, right? So I can see that a lot of you are here already. Good evening and welcome to the class, and thank you so much for being such wonderful and amazing students. I do understand that we had to reschedule it a little and push it back a little because of the previous session, but thank you so very much for being so understanding, for being so amazing, and for being ever excited as usual, right? So welcome, good evening, yes, lots of love to all of you, right? So very quickly before I get started, I'm not going to waste too much time in the introduction. I hope my audio, my video and my screen, right, and what I'm writing on the screen is visible to all of you. Yes, is it visible? Are we clear? I want to check two slides, okay, because we have one slide which has a dark theme and there's a slide which comes much later on in tissues which has a lighter theme. So we will check our technical things in the beginning and then I'm going to get started. Yes, this is visible. All good to go? Hi ma'am, please start quick. Arey, I will start for sure once you tell me that slide सब ठीक है, right? You're able to see. वरना कुछ देर बाद आप बोलोगे कि मैम कुछ नहीं दिख रहा है मेरे को स्लाइड में, आप एक और बार लिख लो, right? Lots of issues will come then. हाँ, okay. So one very quick thing, I want you to tell me if this slide is visible. Let's keep this. So Ravi, let's just check this slide also with the students and that if it is going to be visible for them. So this will come in tissues, okay? I'm just keeping this slide so that you can see. Is this also visible for all of you? On the screen, right? All clear? You are able to read, everything is legible? Lighting, everything, because later on Ravi sir, if he's not there and you say, ma'am, dekh nahi raha, you shouldn't be like, what to do, no? Okay, brilliant, brilliant. All good. Then let's get started. Yes? I'll go back up to the beginning, okay? So now, of course, we're getting started with the complete biology foundation class that is there. So this will help you quickly revise your chapters, help you for preparing ANTHE. For those of you who have already registered for ANTHE, right? I hope that you've registered. And here's a quick reminder for you. So this, especially for 9th grade students, right? These are some spoken English classes that we are providing. It is available for 8th grade and 9th grade. And see, this is not a free program. This is a paid confident speaking program, but definitely it will help you, right? It will give you confidence to speak in public, Cambridge certified teachers. You will have full access to BYJU'S English app. So for those of you who may struggle a little with English, this will definitely help you. And again, a quick reminder, this is a paid course, of course. So we're telling you, if it's something you're interested, try it out, okay? That's all on your discretion. And again, this is as part of Foundation Fest for ANTHE, that's Akash National Talent Hunt exam. Register, enroll for free. You know it's happening from 5th, right? So online exams are from 5th, offline from 6th. Do not forget to register. Link is there in the description box. So now, of course, as you know, in today's class, we are looking first at Fundamental Unit of Life. So this was the first chapter that you had learned quite some time back. So we are going to recall the chapter. Then we will learn and look at the chapter Tissues. And then this is a chapter that we've done very recently, that is Improvement in Food Resources, ending with a Menti quiz. Yes? So here's a quick mind map of all the concepts that we are going to be covering in cell structure, fundamental unit of life, right? So this mind map, I would always tell you when you are studying, right? When you are studying for your exams and you are preparing, I will always tell you to have a mind map first, or keep the mind map with you. You can take the screenshot. I will also be sharing this PDF as well, right? So here you have cell history, you have cell functions, then you have types of cells, and this is all that we're going to discuss in detail. Types of organisms based on the number of cells, cell components. Then of course we also have in cell components, we're going to be learning about cell membrane, about the transport in cell membrane, right? How different types of transport takes place. We look at cytoplasm, cell organelles, all of these organelles we are going to be discussing. Yes? So this is a quick mind map for all of you, right? And we're going to get started with this. Do you want to take a quick screenshot? Let me know. Do you want a screenshot? Yes? Okay. All right. So let me go back. You can take a screenshot of the previous one and I'm going to come back to this. Okay? Take a screenshot and I'm going to come back. Very good. Okay. All right. So I hope all of you have taken a screenshot, right? Have you taken it? Give me a thumbs up so that I can go ahead. I'll send the PDF also. Don't worry. Okay. I'll send the PDF. Ma'am, lesson 3 is not in ANTHE. It is not there in ANTHE, but of course it's good to revise lesson 3, right? Yes. And for those of you, Himanshi, Honey, who are all asking about Ankita ma'am is actually on leave. That's why I'll be taking these classes. Okay. Yes, all right. Go ahead. Yes, going ahead to the next set of things. You can take a screenshot and I'm going to start the class. All right. Let's get started. How long is this session? This session will go on for about 2 hours to 3 hours, okay? This is a marathon class, so I hope you are all prepared. Yes? And where can we find the PDF? You will be able to find the PDF on our Telegram channel. Yes? All right. Okay, so let's get started. All right. So now we're going to get started. Oops, I missed nucleus. So first and foremost, let's start with fundamental unit of life, right? So the fundamental unit of life is nothing but your cell. So why not we calling this chapter as cell, but why are we calling this chapter as fundamental unit of life? What is the reason behind it? Now the reason being that all living organisms who are there, right? All living organisms are made up of cells. So we say cell is the fundamental unit of life, or what we simply say as the basic unit of life. Now this is a simpler definition of cell. But how do we define a cell? If cell is so basic and this is what makes up every living organism, how do we define cell? This is the definition. Cell can be defined as the basic structural and functional unit of life, or you can say of all living organisms. Now, as biology students, you should know why we call it, or why these terms structural and functional are key points, right? So why do we say it is the structural and functional unit? Now, if you take any living organism, so let's take a simple living organism like a leaf. Yes? So let's take the example of, okay, all right, just give me a moment. So let's say we are taking a living organism like a leaf, okay? Now, if you take a leaf, we know that this is how a leaf would look like. Now, if you look closely and you look at its structure and you zoom into it, you see that they are made up of many cells like this. So many cells are coming together and they are what is giving the structure for the leaf. Hence this key term, structural unit of life. Next we have functional unit of life. Now what do we mean by a functional unit? We know that what is the role of leaves? Leaves are there to perform photosynthesis, right? This is one of the most important point about the leaf. So they perform photosynthesis. But when I say leaves are the ones performing photosynthesis, who is exactly doing this process? They are nothing but each and every cell. The cells that make up the leaves are the ones who are doing the process of photosynthesis, which means it also does some functions, right? So it forms the functional unit. So are we clear on why we say it is the structural and functional unit? So sometimes they may ask you to explain this also. This is a higher order, it's a slightly conceptual based question if they ask you why it is known as structural and functional unit. Yes? Very good. It is all clear. You will get the session PDF. Don't worry. Okay. Telegram link in the description box. Yes. All right. Now, of course, moving on, we will look at cell and its discovery. So now we know that it is the basic unit of life, right? And it is a structural and functional unit. But did we always know that cells were the basic unit of life? Not really, okay? Lot of research was done, like way back in 1600s and 1700s. Scientists were super curious as to what is going on within our body, and we know that there were various scientists who were involved in this, okay? But the first who coined the term cell, right, who was nothing but Robert Hooke, right? So you need to know Robert Hooke was the one who discovered these cells, right? Now, can you tell me what was he observing? What was Robert Hooke observing? In the chat, can you tell me? What was he observing? Which specimen was he observing? Very good. In 1665, he discovered it. No, what did he observe? He observed cork cells, right? So he took a slice of cork. Now, what is cork? Cork is nothing but the outermost layer. In bark of tree, you will find cork cells, no? And are these cork cells living or dead? Can you tell me in the chat? Are cork cells living or dead? Very good, very good. They are dead cells, right? So we know that he observed dead cells. And as you are all telling me, when he observed it, yes? So when he observed it, he saw that these cells were looking something like this, okay? Now this reminded him of a honeycomb-like structure. So honeycomb is in the hive of the bees, right? And it reminded him of a honeycomb-like structure. In some cases, he also tells that it reminded him of the rooms in a monastery or the cells in a monastery, which is why he gave the term cell, right? Due to its appearance, due to their box-like appearance or honeycomb-like structure that he observed, yes? So the term cell actually comes from the term cella, right? Which means it talks about those rooms in those monasteries. Hence the name cell. But apart from that, right, it also, very good, it reminded me of the small compartments that are there. Yes, very good. Cells in which monks lived. Very good, very good. So this is what we observe, right? But we also have some other scientists who we need to know. That is Anton van Leeuwenhoek. Now, I'll tell you an interesting fact about Anton van Leeuwenhoek, okay? Anton van Leeuwenhoek was never really a scientist per se. He was actually a commercial businessman. He used to be in the textile business. And Anton van Leeuwenhoek was very curious. Like, he was in textile business, but he was always very curious on what was, like, he's selling cloth. But what is making up his cloth, right? How does it look like? What does it, if I looked closely, how would it be? And that's how he went on to discover different forms of microscope. And he made various versions of microscopes, okay? But some of the advanced versions of microscope helped him visualize some living cells. See, he was the first to discover the living. Robert Hooke did see cells, but cells were dead. Living cells were observed by Anton van Leeuwenhoek in an improved microscope. So he made various versions of these microscope, right? Now there are two other scientists also who you should know as per your textbook. Yes, I'm just going to make some space here. Who are the two other scientists? Can you tell me in the chat, right? Who are the other two scientists we should know about? Ma'am, when is Menti? Menti is towards the end. Wasn't Leeuwenhoek Robert Hooke's assistant? Not per se. Yes, very good, very good. We have Schleiden and Schwann. Very good. Then you have Robert Brown. Yes. Now, very good, everybody. Before I get to Schleiden and Schwann, can you tell me what was the role of Robert Brown? What did Robert Brown do? What did he discover? Yes? Alexander Fleming is also there, but with respect to history of cells, right? Purkinje, yes. Do not mind my handwriting. Yes, he discovered the nucleus, right? That's what he did. He discovered the nucleus, which is the dark structure that we found or that we observe in the center of the cell. Yes. Now, what about Purkinje? What did he discover? Yes, very good, everybody. Very good. Swati, can I answer this question when I tell you about the components of cell, how we identify cells as living or alive, right? Protoplasm, he coined the term protoplasm, which talks about the living components of the cell. No, protoplasm refers to the living components. Very good, everybody, very good. So in this, as you can see, these are two other scientists you should know about. 1 mark questions can come from them, okay? You should know about their discoveries. Another few prominent scientists we should know about are M.J. Schleiden, Theodor Schwann, and Rudolf Virchow. Now these guys are very, very important to us because they came up with something known as cell theory. And as again I'm telling you, in your higher grades, right? This cell theory will be very important because you will get questions. Sometimes what they will do, no, they will ask you this question for 2 marks. State the cell theory, or what are the postulates of cell theory. Another version of this could be a true or false based question also, or an assertion reason based question. Multiple questions can come from your cell theory. Yes? So what is cell theory? Cell theory is nothing but certain postulates which were put forth by some eminent scientists. They were zoologists, botanists who studied different specimens, and they came to a conclusion that these are some fundamental things about cell, right? So these are some basic things. They are universal things as per them, right, about cells that are there. And what are these postulates? That all living organisms are made up of 1 or more cells, which means all living organisms are made of cells. And cells are the basic unit of life, or they are the basic structural, and I will add into this, it is the basic structural and functional unit, right? So they serve as the basic structural unit, they serve as the basic functional unit. So these two were initially proposed by Schleiden and Schwann, right? Now, later on, Rudolf Virchow came up with the fact that all cells arise from pre-existing cells. That means that at some point, if life had to start, it started out as a single cell. And this went on to give us multiple cells that are there, right? So this is very, very important, yes? Universal truth I will not say because we are still discovering a lot of things, right? So I will never say universal truth because there is so much more scope to understand more about life, right? Very good. By the way, this is not okay. All right. Can we all focus on the class? Because I don't know if you are having some other conversations in the chat, okay? Crystal clear. All right, fine. See, I don't want to take too much of your time. Are we clear so far? Then where was the first cell formed and how? Yes, all right. So I'll tell you about that. I think I have it here. Yes. See, around 3.8 billion years ago was when the first cell was formed. And this is not hard and fast rule. This is what they believed. And the interesting thing was what they observed, no, was that there were many inorganic components. Okay, so various organic and inorganic components, they came together and they made up a cell. So as we learn more about it, you will realize that the components inside the cell are nothing but some, organic compounds. They might be like nucleic acids, carbohydrates, or they'll be proteins, which if I pick it up individually, no, they'll not be able to survive by itself. But somehow they have all come together and they formed something which was living. And that was the first cell. And this first cell over time evolved and evolved to give you your ancient bacteria or your archaebacteria. Bacteria are super old, okay? They are ancient. They are like very, very old. I'm not able to think of any old reference, but super old. And of course, your ancient bacteria gave rise to the modern bacteria. It went on to giving you more organisms which are there, so more of them were able to survive. Then you see more of like algae, then your advanced cells that are there came into picture. So your cells have not been something that just came up one day and then you have all these organisms that you see. It has evolved over a period of time, right? So for those of you who are asking me about this, no, this is very, very important. Yes, all right. Yes, probiotic. Very good. Yes, Hari, ask me whatever doubts you have so far. Any doubts? Are we clear? Especially, I know one place where you would have had a doubt was the structural functional unit part. But are we clear with that? Yes, all right. Very good. Was the first cell, ma'am, the first cell was animal or plant cell? See, the first cell we'll not categorize as animal and plant. Animal and plant cells are very advanced cells, okay? And as we look at the structure, we will understand that they're very complex and advanced cells which have been formed much later. The first cell would be a very primitive cell with a very basic structure like how a bacteria would have, but even a simplified version of that, okay? Yes, no doubts. Let's get moving, okay? All right, let's have a look. So now that we know all about cells, right? We know that, Yes. So now that we know all about what cells are, we know what are cells, we know that all living organisms are made up of cells. Now there are different kinds of cells that are there, right? So you see that there are some organisms and we call them as organisms. Now why am I using this word organism? What is the reason behind it? Now organism is when I'm talking about a living entity, and I know you will ask me this, ma'am, what is organism? So an organism is a living entity that can survive by itself, right? Now an organism can be made up of just one single cell, or they can be made up of many cells. So we see that organisms can broadly be of two kinds. So types of organisms, I would say, is what I want you to focus. So how am I coming to this? Why am I talking about types of organisms? Because at the end of the day, living things or living organisms are made up of cells, no? So because of that, you have two kinds. You have unicellular and you have multicellular organisms. So what are unicellular? Uni means one, right? Very simple and easy. Multi means many. That means they are made up of many cells. So unicellular organisms are those wherein a whole organism is made up of just one single cell, right? And this cell has the ability to carry out all the functions as an organism. So it can do everything that we do. So if we need food, a single celled organism will also be able to do that, right? So here we have some examples like amoeba, bacteria and paramecium, but in the chat, can you give me some more examples? Can you give me some more examples of unicellular? Ma'am, amoeba is an organism or a microorganism? Amoeba is a microorganism because organism is a living entity, okay? So amoeba is an organism, but because I cannot see it with my naked eye and I require a microscope, we call it as a microorganism, yes? So to be specific, we say microorganism, but you know, organism is also correct. And most often than not, unicellular organisms are microscopic. Very good. Euglena, I can see yeast, I can see Chlamydomonas, yes. Probiont is like the initial cell that was there, right? Very good, very good. Algae is very vague, okay? Because algae can be uni or multicellular. So algae will be a very vague, example to give for those of you who are telling me algae. Plasmodium, yes, very good. So you all know what is unicellular organism, no? Then you have multicellular organisms which are made up of many cells, right? And all these cells will come together and they make up the whole organism. And in this, because they're made up of many cells, we will observe body organization, right? So humans, plants, fungi are all some examples. Now quick question. Multicellular organisms will have the same kind of cells or they will have different kinds of cells. Or I'll make it easier because that can be a vague one. Between plants and animals, will we all have same kind of cells or different kinds of cells? Yes? Virus living होगा या non-living? Virus is a virus वो in between है. We see that there are some characteristics which have living, they observe certain living characteristics, while in some they don't show those living characteristics. So we'll not really say virus is living per se, right? Yes, all right. They are made up of different cells. No, we don't find the same kind of cells. Yes. Now, how are they different? Can you tell me? How are these cells different from one another? Yes. For those of you who are asking me again, Menti will be towards the end after I recall 3 chapters. All right. Living and dead, okay. Shape and size, very good. In shape, yes. Division of labor, very good, very good. So basically they come in different shapes, they come in different sizes, right? And some of you are also telling me sometimes they might be living or dead. Yes, very good. So in this case, if you see, we know that there are various types of cells. I'll not say function, but I'll say based on shape and size. We know that some cells are very small, some cells are really long, some cells are, really big, right? But the shape and size, or mainly I would say the shape of the cell, is in such a way that they can carry out their function effectively. Now you will definitely get this question, right, either as a true or false, or you will get it as a 1 or 2 marker on why do different cells have different shapes, right? What is the reason behind it? Simple. To carry out their function effectively. So if you have a RBC are red blood cells which are biconcave in nature, right? So we know that they are let me just erase that out. We see that they are biconcave, right? Or I can say that they are discoid in shape. They have a disc-like shape. Why? To carry out their function, that is to transport your oxygen. Or why is a neuron elongated, right? What is the reason behind a neuron having an elongated structure? So that it can help in transmission of impulses. So when you're writing this answer, you can give an example as well to support your answer. Are we clear? Very good. To transport, they have hemoglobin, they have, help in transmission. Yes. Smallest cell in the world will be mycoplasma, right? It's extremely tiny. Mycoplasma is the smallest. Yes, all right, very good. Atoms are smaller or cells? I'm not sure of this, but I think atoms, right? Yes, all right. Okay, now moving on, right? So we know that there are different cells which are in different shapes and different sizes. Can you repeat this once again? Of course I will. See, we have different cells which come in different shapes, right, and different sizes, okay? Now, why do different cells have different shapes? They have different shapes so that they can carry out their function effectively. So we know that an RBC is biconcave in nature because it helps in transporting oxygen. So what happens is that this disc-like structure that is there, it helps in transporting more amount of oxygen, and it also helps in the movement through the blood vessels. So imagine if the RBCs were really huge, right? We see that very less RBCs would be there and will help in transport. But this particular shape will accommodate more RBCs in the blood, right? Now they can all get arranged and more RBCs can be there so that they can flow through our blood. And as a matter of fact, we have almost 5 million RBCs in our body. So you imagine, if they were huge spheres of balls, it will be very difficult for it to flow through our body and it cannot accommodate 5 million. But this shape allows it to be more efficient. That is what you need to understand. Are we clear? Yes, all right, very good. Our atoms, after being in class, in chemistry class, you should not be asking our atom cells, right? We know what atoms are, yes? Okay. Are we clear so far? Very good. Lifespan is 120 days of RBC. Everybody who is, asking me, ma'am, please do Menti, I will do Menti. I'm just going to revise all of these topics and then we will get to Menti. Okay, I need you all to be very patient in class. I want you to be attentive in class. Okay? Yes, all right. Ma'am, biconcave, it means it will be like this. See, this is what is biconcave. All right, and a lot of K-pop conversations. I would like all of you also to focus in class, okay? Yes, Swati, you are right. Yes, you are. So now that we know there are different shapes, there are different sizes of cells, all are performing effective functions, we need to know what functions a cell performs in general, right? Because a single cell, there are some organisms which are just a single cell and they are able to carry out the function. So what are these functions a cell performs? So these are some basic functions of the cell, okay, which all cells will carry out. Yes? So cells can obtain nutrition, right, which means that they can take up the nutrients. They convert it to energy, which means energy production happens within cells. They get rid of waste material. They create copies of themselves, or they make more, or they reproduce, right? And when you go to 10th standard, you will realize that these are all some life processes that takes place, which are all happening inside the cell. So activities inside the cell is effectively what is making us, it is helping us survive. Yes? Very good. Is the syllabus of NCERT, CBSE same? Okay. So now let's quickly go on to understanding more about who is carrying out these activities, right? So we know cell is there, cell is doing life processes or doing all these activities, but who exactly is doing it? Can you tell me in the chat? Who is doing all of this inside the cell? In the chat very quickly. Yes, very good everybody. I'm getting the answers. I can see Chahek, Hari, Nirmala. Yes, very good. Varshika. Okay. See, for those of you who are telling me specifically mitochondria, ribosome, nucleus, you are right, but you are only partially right. Because ribosome alone or mitochondria alone or your nucleus alone will not be able to keep the cell alive. But all of them together, which form the cell organelles, is what keeps the cell alive, right? So you have cell organelle, you have your components like your cell membrane, nucleus, cytoplasm, mitochondria, endoplasmic reticulum, Golgi, lysosomes. So we see these in animal cells. Then of course we have plant cells which have some extra cell organelles, which are nothing but your cell wall. It has vacuoles and it has plastids. But at the end of the day, ma'am, what are cell organelles? What are they? So we see that cell organelles are nothing but they are little organs, okay? Which means that they have a definite structure, a definite function, and they are what keeps us alive. They carry out these activities, which is why we call them as little organelles, okay? So this was a quick introduction to some basics of cell. Yes, are we clear? Are we clear? Ma'am, how does it convert it to energy? Can I tell you this when I explain mitochondria, netra? Yes? Ma'am, अगर hemoglobin में iron है, water and oxygen present है, तो उसमें rust क्यों नहीं होता? Because of the chemical nature, right? In chemistry, chemical reactions takes place only when they are of a certain nature. So your water is not just hydrogen and oxygen, right? And you know that even in your oxygen, it's not that it can just freely react there. So that's why you don't have rust formation. Yes, okay. So now of course we are going to look at the structure of the cell, okay? Now can you tell me what are the three basic parts or the basic components? What are the three basic components of every cell? And here I am talking about some advanced cells, okay? Don't tell me bacteria. What are the three components? Can you tell me? How many chapters will you revise? 3. Very good. I can see the answers. No aspirant, not electron, proton, neuron. That is in your chemistry. Yes, we're talking. That is when you, with respect to a certain structure, right? But here it will be cell membrane or your plasma membrane. Then you have told me nucleus. Yes. And then you guys have also told me cytoplasm, right? Very good. Abdullah Razi, what do you want me to explain once again? Can you tell me? Yes? Quickly, let me know what is what. Wonderful, everybody, wonderful. So these are your 3 basic components, right? So now let's quickly look at the components of the cell, starting with the cell membrane. Now see, I'm going to spend some time here because in cell membrane, you need to have certain understanding of transport, places where you will definitely get confused. So I'm going to spend some time here, and whatever doubts you have, please get it clarified. I want conceptual clarity to happen in this class. Yes? So let's focus, okay? So cell membrane, if you look at the structure, the cell membrane is the outermost boundary, right? So we see that it is the outermost boundary of the cell. Or in plant cells, I will revise and say it is the outer boundary, right? Now what does the cell do? The cell membrane do per se? The cell membrane that is there, it will give the cell a certain structure, right? So it will enclose all the cellular components. What do I mean by it will enclose the cellular components? It will make sure that all the things which are there inside the cell, no, it stays where it is, so that it doesn't wander off outside or it starts leaking or it spreads everywhere, right? So it provides a boundary to the cellular components. Now secondly, apart from this, what we also observe is that it is made up of a phospholipid bilayer. Now very, very fancy sounding thing, phospholipid bilayer or lipid bilayer. What do I mean by that? I will not say lipid bilayer because I find it incomplete, which is why I will always say phospholipid bilayer. Now chat pe I know some of you will say, ma'am, what is this? Why are you saying extra things to me? Why do I need to know this? It's good for you to understand because they have vaguely mentioned it in NCERT. That's why I want, if you're learning it, learn completely. Don't learn other information. Learn fully so that you know what it is, and later on you will not have a misconception, right? Yes. Are there nuclear reactions? No, there are chemical reactions. I'll not say nuclear, okay? For Swastik, who is asking me. All right, what is in there and in between? I will tell you, right? Not understanding, ma'am. Hey, no problem. I am explaining, Tina. I will explain. Okay. So what is the difference between, I got a very interesting question, I think from Himani, right? What is the difference between phospholipid and only lipid? See, lipid is actually only going to represent one part of it, okay? That is the lipid tail that is there. But actually, in this structure, you have a phosphate head also, right? So this is why we say it has a phosphate head. So I'll say it has a phosphate head and it has a lipid tail, right? Which is why phospholipid, this together is making it. And why am I saying bilayer? There are two layers of it, hence bilayer. Now, can you all tell me what do you find interesting about this arrangement in the chat? Can you tell me what is so interesting about this, right? What is interesting about the arrangement that you are seeing on screen? Yes, Srinivas, yes. Okay, fluid mosaic model. Okay, Mahendra, don't worry, I'll explain this whole thing once again. Yes? Okay, very good. Fluid mosaic model, it is sandwiched. Okay, upper layer, hydrophobic, hydrophilic. Very good, very good. I'm seeing some answers coming in. Very good. So I'm going to maybe switch to another screen to show you this clearly. See, some of you are asking me, ma'am, what is phosphate and what is phosphorus? Phosphorus is like this, right? Phosphate is your PO4. I am not doing the valency part, but this is the difference between your phosphorus and phosphate. So now you know the difference, right? So are we clear with the difference between phosphorus and phosphate? I'm leaving it on screen for you all to understand better. Now what happens is that your cell membrane is arranged with many such phospholipids, okay? Now this part is the lipid tail that is there, right? So this is the lipid tail and this is your phosphate head. Now lipids are nothing but a kind of fat, right? Now we know that fat, and the most common example of fat is nothing but oil, right? It is a very common example. Fat and oil and water don't mix. And we know that most of our body, around 75%, is made up of water, which is why the cell membrane is arranged in this manner so that the lipid tail is on the inside. This is your water-hating part, right? This hates water. It cannot mix with water. So it is on the inside. While your phosphate part has no problem with water. So this is your water-loving part. For those of you who are telling me hydrophobic, hydrophilic. Phobia means I don't like it, I'm scared of it. So the lipid part is hydrophobic while this part is hydrophilic. This is some extra information for all of you, right? Hydrophilic head and hydrophobic tail. Yes, all right. Very good. So this is the structure and they are made up of many such phospholipids arranged. Are we clear, everybody? Are we clear? I'm going to explain once again. You have a phosphate head, a lipid tail like this. It looks like a human being. The head likes water, tail does not like water. Tail is your lipid tail, head is your phosphate that is there, right? It is your phosphate head. Okay, so this is what we mean by it. Are we clear in the chat? Give me a quick thumbs up so far. Are we clear with the structure of what do we mean by phospholipid and why we say it is a bilayer? Because there are two layers arranged in this manner. Yes? Are there parts in the human body that get oxygen directly from the air and not from the blood? No. Mainly it's transported through the blood only. Yes, all right. It's clear, ma'am. Okay, very good, very good. Yes. Now, of course, if you see, apart from this bilayer, you also have some proteins, you have some carbohydrates which will be arranged, right? And they're all arranged and the way they are arranged will give you like a mosaic pattern, which is why it's called as fluid mosaic model. Why do we say fluid mosaic? Because some of you are asking me, and this is extra information, don't get worried, it's not there in your syllabus, or if they ask you also, it's a 1 marker. Why do we say fluid mosaic model? Because of the arrangement of your phospholipid bilayer along with your proteins and your carbohydrates is what we understand as phospholipid bilayer, right? So now, of course, apart from this, yes, we know that it's giving you the boundary, this is the structure, and most importantly, we know that this cell membrane separates the inner part, right? It separates the inner part of the cell from the outside. So that's why we also call it as a boundary. Now, when we talk about the function, right, we know that this is very, very important. The main function of the cell is to allow or regulate. See how I'm using the words here. See the words, it's been used. It allows, that means permission is required. Allows what? Entry and exit of some materials in and out of the cell. So it regulates the entry and exit of substances. And because only selected substances are allowed to enter into the cell, we call it as selectively permeable, or we say semi-permeable, right? Both are correct. So they are selectively or semi-permeable. Yes? So this is the main thing you need to remember, and they also play a key role in transport, right? So all this entry and exit is nothing but transport. Yes? Everybody, give me a quick thumbs up. Yes? What is nature of existence? Okay, all right. So I think Swastik, can I answer this in some time? Yes, okay. So now very quickly I have a doubt which is coming in, difference between cell and cell membrane. Cell is nothing but the basic unit of life, right? Cell membrane that is there is nothing but a component of this cell. Yes, it is a part of the cell. Are we clear? Yes, yes, thank you Srinivas. I will cover a major part and we'll take a water break together, okay? All right. Himanshi, selectively, see, at a higher level you will understand that selectively and semi-permeable are not quite the same. But at this point you need to understand that selectively or semi-permeable is more or less. Semi means it will allow only certain substances to pass through. Selectively because it can select and send it through, right? So in the case of your cell membrane, we say selectively permeable because it has the ability to regulate it. But there are like some, things, like for example, if you see when we learn about osmosis later, we see that there are some sheets, like for example, like muslin cloth, or you can use a potato, where they're just semi-permeable because of the structure of it. It will only allow certain to pass through. That is the difference between selectively and semi-permeable, okay? What are antigens? Antigens are nothing but structures which are found on the surface of the cell that in our case, for example, helps us identify if whether this particular thing, right, whether this particular cell belongs to our body or it does not belong to our body. No? Okay. Very good everybody, very good. So now we will move on. Now here we have learnt about transport. Now when we talk about transport can happen in various ways, no? And this is the most scary part in this chapter. Now transport can happen in two ways. For transport to happen, one is it can happen by taking or consuming energy. So if I want to run from here to there, I want to move, I want to transport myself, I need energy. But sometimes I don't need energy, right? So there are some transports of substances which do not require energy. But before I tell you what requires energy and what doesn't require energy, what are we transporting? Like what is going to the cell? What is getting transported to the cell? So what are the substances which are getting transported? Can you tell me in the chat? Very good, very good. Yes, active and passive. Yes. So active passive is what we call. So if it requires energy, we call this transport as active transport. If it doesn't take up energy, we call it as passive transport. But what substances need to get transported? Very good. Yes, very good. Water needs to be transported. Okay. Then what else is there? Nutrients. Yes. Nutrients need to be transported. Food needs to be, okay, nutrients, food is covered in that. Sap, okay. Energy, transport, okay, fine. Biomolecules, okay. Yes, finally somebody told me my gases, right? Oxygen and your carbon dioxide need to be transported. No? So you have all of this, right? Okay, White, what is the problem? It is class time, so please focus. Yes? Okay, very good, very good. Ions, yes, I was waiting for this also. Like your calcium, your sodium, your potassium. Yes, all of this is required. Yes, all right, very good. So now I do see a lot of doubts coming in, okay, some interesting doubts are there. So I would request all of you, if possible, right, please write your doubts down, because when I'm doing this part, I want all of your focus here. And once I finish this, I will take all the doubts at one go. Can you give me a quick thumbs up for that? Yes? Give me a quick thumbs up. Yes? Are we clear? For those of you who are asking, ma'am, I have all these doubts. I'll finish this part because this is the most important part of the chapter, and then I will do. Yes? Thank you so much. Brilliant, brilliant students. So now very quickly to recall types of transport. Active transport is there, passive transport is there. But at the end of the day, what is getting transported? Your water, nutrient, gases, and your ions. Now, first and foremost, let's talk about this scenario, right? So sometimes, I'm sure when we have a very fragrant, we have fragrant tea, or sometimes, when there's something that smells really good, it starts at one point, right? Or I'm sure when at home, when your mom was making sweets, at home everybody would have had sweets for Diwali, no? Mummy will be nicely making in the kitchen, but when you're sitting in your room, after some time you will be able to smell, all the yummy, yummy, delicious sweets that you had, right? So how is this happening? By a process known as diffusion, right? So how do we define diffusion? So we define diffusion simply as and write this down, this is very important. So diffusion is the free movement, or another term you can use is the spontaneous movement, right? So we see that it is the free movement or the spontaneous movement of substances. Now from where to where is it going, right? It is going from a region of higher concentration to lower concentration, okay? To a region of lower concentration. Now this is very important. You might get to define the process of diffusion, right? So take a quick screenshot, everybody. I know my handwriting is not very great, but you can have a screenshot as well, right? Now what do we mean by spontaneous? Immediate, right? That means it's immediately getting transported. It's not waiting for 5-10 minutes and then moving. It's happening almost instantaneously, right? Yes. Now I'm getting another question, ma'am, what is this spontaneous, right? So very quickly here, if you see, what do we mean by concentration? Concentration is the amount, right? So here we are talking about how much of it, right? What is the amount that is present? That is what we mean by the term concentration. Now we know diffusion is where they move from higher amount to lower amount, right? And we are not mentioning any semi-permeable membrane, nothing of that sort. It is just free movement. So where does this free movement occur? Where does this diffusion occur in our body, and why are we learning about it? If my mummy is making sweets in the kitchen and I am able to smell it in the room, that's because of diffusion. Okay, molecules are they're coming all the way, they're freely moving, right? But how has it got to do with anything what's happening in my cell? Simply because if this is your cell, right, we see that transport of gases, right? So transport of your gases like oxygen and carbon dioxide takes place by this very process of diffusion. Yes? So here in this case, we see that oxygen, which is present in more amount. You can see that some of you are telling me, ma'am, this is there in chemistry. Yes, chemistry in bio. When this oxygen is present, it will be present more in amount outside the cell, because oxygen is present as a gas, we take it in, then blood will transport it, and finally the blood will be bringing the oxygen. So definitely more oxygen in the outside than inside the cell. So inside the cell maybe little will be there. So what will happen? Due to difference in concentration, from higher amount it will move into lower amount. Vice versa, we know that carbon dioxide that is there, no? Carbon dioxide is given out as a byproduct of respiration. You've learned this in 7th grade. So more amount of carbon dioxide inside the cell, but the blood will have very little carbon dioxide because in our surroundings not that much carbon dioxide is there. So when we breathe in also, very little carbon dioxide will be taken by the blood. But we also know that from the cell it will go out in this manner, and both of these processes, right, is happening by diffusion. So are we clear on diffusion, everybody? Are we clear? Yes, everybody, very quickly, are we clear? Ma'am, what is the difference between diffusion and osmosis? I will be telling you, okay? All right. Okay, I can see some of the chat is getting distracted. So please, everybody, please don't get distracted in the chat. Please focus on what's happening in class. Okay, I'm very clear about this. Ma'am, which type of substance usually undergoes diffusion? We observe mainly with gaseous substances, right? Your gaseous substances tend to show this free molecules. Okay. All right. So for those of you who are asking me for the definition, this here is there on screen. Okay, this is there on screen. I'll start Menti after I finish 2 more chapters. I'm covering the main parts. So what we will do, we will try to aim to wind up fundamental unit of life by 7:45. Okay, so we'll try to dedicate at least 45 minutes for a chapter to thoroughly revise it, and then we will do Menti. That is my plan. All right. So now we will move on to the next one, which is, so like I told you, this is already there on screen for you as well, how movement of oxygen takes place. Now next one is a very important one where you can get a lot of application and reasoning based questions. That is oxygen. That is osmosis. Sorry. So now you know how your gases are transported, right? So you know how they go. Now you have your osmosis, okay? Now osmosis is a process by which your water is transported in and out of the cell, okay? So how do we define osmosis? This is very, very important, and I'm going to write this down. So osmosis can be defined as the movement. So it is defined as the movement of water molecules, and I will also write in brackets solvent, okay? So it is defined as a movement of solvent molecules or water molecules from a region of its high concentration to a region of its low concentration, same as before, but one addition, but across a semi-permeable membrane, okay? Very quickly, write this definition down or take a screenshot. You will get this, okay? This definition can come. They'll ask you for 1 mark, or you will have questions based on this definition. Take a screenshot, everybody. Very good, Netran. Very good. Yes, all right, everybody, all right. Okay, not understood, ma'am. Okay, can you repeat semi-permeable membrane once again? Okay, took it. All right, ma'am, what to do with definition? Take a screenshot. That's it. You don't need, because after I wipe this off, it might not come back. Okay. So now, for those of you who are asking me, ma'am, I did not understand. See, we also know that water that is there is required by the cell, right? So what happens is that based on the water concentration, okay, that I will show you with another slide. See, Your cell membrane is there, no? Your cell membrane is semi-permeable. Some of you are asking me, ma'am, how can it be selectively permeable? I will tell you. You have semi-permeable cell membrane. So what happens is on the outside, if it is higher, right, we see that water molecules from the outside will enter. But here the semi-permeable membrane is the condition for osmosis to happen. For diffusion, semi-permeable membrane is there or not there, it doesn't matter. But for osmosis, this is very, very important that from higher concentration, it goes to lower concentration. Yes? Are we clear? Are we clear? Ma'am, diffusion also occurs in semi-permeable membrane. Exactly my point. But there, it doesn't matter. So if I remove the semi-permeable membrane also, no, it will still happen. But in the case of water molecules for osmosis as a process to happen, this is the key thing to be there. Yes? Are we clear? Very good. Ma'am, moment water will continue till concentration. Yes. So osmosis will continue till in both sides it becomes equal. Even in diffusion, as a matter of fact, the process will keep happening till equilibrium is reached. See, at the end of the day in chemistry also you learn equilibrium is something that we need to attain, right? Yes, all right. Ma'am, what is this hair? It's not hair, okay? It is lipid bilayer. This is not hair. Okay, all right. So what does semi-permeable membrane mean? It's a membrane that allows only certain substances to pass through. Okay? Yes, all right. So now of course, we know that based on the concentration of water, right, movement of water can take place. So one is when on the outside it is more, water from outside will come into inside. But there's also a possibility that if a cell is placed in an environment where there is more water on the inside and less on the outside, then it can move out also. So it is not a one-way thing. Even when we learnt about diffusion, it's not only outside to inside. It can be inside to outside also. It's in and out. So here, based on that, right, based on this movement, we know that there are three types of solutions which are there. And these three types of solutions, based on their composition, right, their chemical nature, we know that certain types of, movement of water takes place, right? So what are the three types on the chat? Tell me very quickly. What are the three types that are there? Yes? Ma'am, please define it. I will definitely define it. Yes, okay. Ma'am, hair-like is hydrophobic. Yes, Shivansh, you are correct. Very good. From inside cell, where will water come? I'll tell you, math science, what will happen. Yes, Riya, yes, all right, very good. Hypertonic, hypertonic and isotonic, okay? So let's start with the simpler one, okay, that is iso. Iso means it is the same, right? So iso means it is in such a way that if I have a solution, okay, so a solution is what? Again, some chemistry coming into the picture. Solution is something which has both solute and solvent, right? So this is what you mean by solution. Now here, the solution is in such a way that how much ever solute is there on the outside, right? Or the concentration of solute and solvent is same or is equal to what is there inside the cells. Which means that if it is same, will osmosis occur? Is there a possibility of osmosis occurring? In isotonic solution, will osmosis occur? Yes or no? Yes, very good. It will not occur. No? It will not occur because you know that for osmosis to occur, it should go from high to low. If it is same, nothing is going to happen. So this is a solution wherein equal concentration of solute as the cell. Okay, so same, inside, outside, no osmosis, equilibrium is there. But now what happens is that if we take a hypotonic solution, okay, what is hypo? Hypo means less, right? So here we see that it has less, okay? That means that the solute, it has less solute in your solution, right? So there is less solute and more solvent. Now how am I saying this is less and more? How do I know this is less solute? I say less and more with respect to something, right? So this is less solute with respect to your cells, right? That means that the water that is there. So I'm going to switch to another screen. In this case, what happens is that more amount of water on the outside, right, when compared to what is on the inside. So less solute, more solute. More water, less water. So water will enter in. Are we clear with this? And if the water enters, this will swell up. So when in a hypotonic solution, we see that cell will swell. Yes? Hypo means cell will swell. Very simple and easy, right? Are we clear? Yes. Okay, everybody. Are we clear? Who may? Okay, Aishwarya Ma'am, please. It is not, I'm not able to understand. Suresh, do you want me to, is it because of the language that you are not able to understand? Is it because you want me to teach in Hindi? Or is it because there are some points that are not able to, you're not able to understand, right? Okay, all right. Now this is what happens in your hypotonic solution, right? So if it is lower concentration of solute, cell will swell. Now what about in hypertonic, okay? So in hypertonic, what is happening? Hyper means more, right? So more solute outside. So there is more solute outside. That means my solvent or my water is very, very little on the outside. But now what's happening? Compared to my outside, my cell is having more water. And again, if osmosis were to happen in this case, what will happen? Water will move out. And if water moves out, your cell will shrink. So more, if it is a hypertonic, cell is going to shrink, right? Yes, okay. Ma'am, explain it once again. Oh, all right, everybody, I'm going to revise this whole thing once again. So you have 3 types of solutions. I know this is the part where you'll get confused. That's why I'm going slow here. Otherwise, I would have moved a little faster. You have hypertonic, okay? You have hypotonic, right? And you have isotonic. Yes, you have these 3. Now what has happened? This is with respect to my solution. So I have a solution, okay? My solution will have what? It will have my solute and it will have my solvent, right? That is what I call as a solution. Now I have taken this and I have put a cell inside it. Now in a hypertonic solution, there will be more solute. Hyper is with respect to solute, right? More solute, less solvent. Now this is in my solution. But what about in my cell? Cell is having more solvent, okay? And it is having less solute. How am I saying more, less, more, less? It is with respect to each other, not with respect to anything random. And then what will happen? You have more, you have less. So in hypertonic solution, water will move out of the cell. If water moves out of the cell will shrink, right? The cell will look shriveled up. So imagine if you had raisins, right? If you've seen raisins, the dried grapes that are there. If you see raisins that are there, you put it in water, or you take swollen raisins and you maybe keep it in salt solution, right? So, or if you would have seen, if you take maybe any vegetable and you keep it in a very highly salty solution, after some time we see that it will shrivel up. It is because of this process, okay? Yes, very good. Now on the other hand, in hypotonic, I'm erasing this, okay? Now in hypotonic solution, I'm going to remove this also to make more place. Now in hypotonic, what happens is that I have more amount of solvent, okay? Very less solute, right? So again, hypo means less, no? So what will happen? I'll put my cell. What is the concentration of my cell, no? Cell is having more solute, right? But less water, or it is having less solvent. So now what is going to happen? Water, more is there outside. So more solvent on the outside, less solvent on the inside. Cell water from outside will go inside by osmosis. So what will happen? Cell will swell, no? So that's what happens in hypotonic. Yes, very good. So now we know this is all with respect to relative concentration. And at the end of the day, we also have your isotonic, but isotonic is so easy peasy, no? Iso means same. So no osmosis is happening. Okay? So quickly take a screenshot of this, because if I move this board, you will not be able to see it again. So take a screenshot and tell me. Okay. Ma'am, can you please give me an example of a freely permeable membrane? Your cell membrane is an example of a freely permeable membrane. Okay? Yes. Okay. All right. Very good. Yes. No change. Very good, everybody. Very good. So now with this, we've learned about types of solution. And now, of course, we will quickly look at plasmolysis as well, because some of you are asking me. See, in plant cells, what happened is that they have an additional layer which we will discuss and which we already know, that is the cell wall, right? So when you put a plant cell in a hypertonic solution where the water is effectively going to move out, we see that they will shrink considerably, okay? All the water will shrink, and because of that all the living components will become, it'll shrink and go to one corner of the cell. So we see that plasmolysis is a condition or is a process where the cell contents will shrink due to the absence of water. It has lost a large amount of water, and we see that at the end of the day it will be shrunk like this, and there will be a lot of space here, and we call such a cell as plasmolyzed cell. This is there in your NCERT, which is why we are stressing on it also. Ma'am, why plant cell will not burst when kept in hypotonic solution? For the very same reason that it has a cell wall. So it has a cell wall, okay? This is actually an interesting one. So when a plant cell is kept inside a hypotonic solution, water will enter inside. So water is coming in, swelling up, cell is swelling, cell is swell. But what happens is that now water is becoming more and more. So what water will do? I need more place, it will be pushing, okay? So it will exert pressure, right? So this is going to exert pressure on the cell wall. But your cell wall, we know, which we will recall once again, your cell wall is rigid. So your cell wall will also be like, no, I'm not going to let you go. So it will also push back. So you imagine if you were stuffed in a small room with like 10,000 people. Everybody will be squashed to one place. There'll be no place to move. Some of them will want to get out also, no? But outside the door, you have big bodyguards. You have like locks. Everybody holding you back saying, no, I am not letting you go, right? So imagine, you've seen this, no? Where people will be like holding hands like this and they'll be like, no, give me place, and they'll be moving. Kind of what happens in a plant cell. So this is what you call as wall pressure, okay? Wall pressure is the pressure which is exerted by the cell wall. Yes? Bone cell, living or dead? Every cell starts off as living only, right? And we learn about bone cells later on. So very quickly, recall diffusion is of course, okay, let me make this a little simpler. So for diffusion, don't write this. Write down that it is the free movement of substances. Okay, write this definition down instead. And here we see that movement is of substances, while here we see only solvent particles move, right? We don't see any solute particles moving. Here no semi-permeable membrane is involved. Here we see that semi-permeable membrane is involved. But what is similar between the two is that both of them happen till the concentration is the same. Yes? So this is the difference between diffusion and osmosis. And in both cases, when we learnt about diffusion and osmosis, we saw that there was no involvement of energy, which is why these two are examples of passive transport. They both don't require energy. But in some cases, like some ions, right, maybe like your potassium ions or your sodium ions, we see that it's not always that they will be in high and low concentration. Sometimes from low concentration to high concentration, they will have to move out. Now, if it is going in the opposite direction, which means I am already very less in number, and then you want to send me out also, right? Cell is like, bro, you want me to send this out? then it should come at an expense, right? You have to give me some currency. And in the case of living organisms, they do it in the form of your energy, right? So at the expense of energy, like large particles and ions require energy to move, which is why we see that there are, of course, active transport that takes place at the expense of energy. Now, of course, if you see endocytosis, exocytosis, of course, exists. But endocytosis, of course, also gives you an example on the flexibility of the cell membrane as well. So here's a quick one of active versus passive. Active transport, molecules move against concentration gradient, right? Or it is in the opposite, from low to high. While in passive transport, this is from high to low, right? That's what I mean by along concentration gradient. Here, no energy is required, energy is required. Sodium, potassium, movement of sodium ions and potassium ions are example. Diffusion and osmosis are examples. Yes? So quickly take a screenshot, everyone. Quickly take a screenshot. Now I can see that a lot of random chat is happening. I want you to stay focused on the chat, okay? Exactly. See here, I can see that some of you are having these conversations, and I would request that you don't have these conversations and stay focused on what we are teaching and learning. Yes? You're all wonderful students, no? So please understand that we are in a class and we are revising. Yes? Crystal clear. Okay. All right. Very good, everybody. Very good. Ma'am, repeat active transport. Active transport takes place when we are going to be. Active transport takes place when ions, right? So normally what happens is to maintain at an ion level also our body has to maintain equilibrium, okay? So what happens is that sometimes to balance out sodium and potassium, sometimes when potassium moves inside, what will happen? There will be less sodium and more sodium here. So what happens is sometimes from low concentration it will go to high concentration. So this movement is what we call as active transport, where it goes from low concentration to high concentration. And at this point what I need you to understand is that for low to high it requires energy. So your sodium potassium pumps are very good examples. Yes? Yes, very good, everybody. Very good. Okay. So now, of course, quickly I'm going to talk about cell wall, right? So cell wall is an additional layer which we find in plant cells. It is made up of cellulose, right? And we know that the major function of the cell wall is to provide rigidity and support, right? This is the main function, and you will get a question which says differentiate between cell wall and cell membrane. That's also another possible question that you will get. Cell wall here is freely permeable, okay? So your cell wall is freely permeable, unlike your cell membrane which is selectively permeable. So this is one quick concept that I want all of you to know, and take a quick water break for 2 minutes. Stretch out your arms, right? We covered some of the tough parts of the chapter, I feel, and the remaining parts are pretty simple only. So very quickly, everybody, I hope that you take a quick water break for all of you. Now, in the meanwhile, I'll just get my bottle of water. Yes, now Arjun and many people who are asking about Menti, I've already told you, I have 2 more chapters to cover in 45 minutes. I'll be faster. I'm only going to go slow in places which on an exam front is very important for you. Only in those places I'll be slow. But other than that, I want you to know that after 3 chapters only, I will be able to do Menti. And I'm trying to be as fast as possible. Okay, all right. Okay, everybody. We don't need break. Very good. Fine. If you need a break, you tell me, okay? But I want you to know, any doubts so far? Yes. Can you say why in potato cubes when we place in water, it becomes firms and increase in size? Due to osmosis. Because I told you, potato cubes that are there act as semi-permeable membrane. Their composition makes them serve as semi-permeable membrane. Hence, water will come in and they will increase in size. Yes? All right. Ma'am, what is membrane biogenesis? I am about to explain that. Yes. So that I will be explaining in just a bit. No problem, Arjun, no problem. Yes, Gaming X, please tell me what is your doubt. I had some more doubts in the beginning, but I think over time, since I've explained the concept, it's been easy. Difference between nucleus and nucleoid. I think I'm about to explain nucleus only. Yeah, so I'll tell you that in just a bit. I'm going to explain. No doubt, everything is crystal clear. Very good. So now let's move on. Plasmolysis once more. Okay, I'll do that one last time. See, plasmolysis is something we will observe in plant cells mainly. Why? Because they have a cell wall, right? Now what happens is that when they are placed in a highly hypertonic solution, what happens is that water will move out. Okay, large amount of water will move, which means everything in the cell will shrink. But the cell membrane, the cell wall is there, which is why what happens is that cell wall is intact, but the rest of them will shrink and form a corner. So we'll have a lot of empty space. This is very different from something shrinking, like an animal cell shrinking. If an animal cell shrinks to that level, it will no longer exist. It would even die, okay? That's why we see plasmolysis is something we observe in plant cells. And it will shrink to a very good extent. And this process is what we call as plasmolysis, and this cell is what we call as a plasmolysed cell. Yes? Okay, yes honey, your chat is visible. It is visible. Okay, ma'am, explain again this part. Okay, if you tell this part, I will not be able to understand. Rasangna, again, if you tell me that which part you are not able to understand, I will help you. But if you tell me, ma'am, I'm not able to understand, then I'll not know which part you are not understanding, no? Ma'am, please explain plasmolysed cell. See, plasmolysed cell, again, like I tell you, you imagine, okay, you have, again, I'll give you an analogy. Imagine you're in a room, right? And what has happened is that they have told everybody. So you have 100 people in a room and they told that at least 90 people have to get out of the room, right? So 90 people have gotten out and only 10 people are remaining. Now what happens is that for these 10 people, imagine they all form a circle in one corner. What will happen? They will all be here in this corner, but there's a lot of empty space, right? And all this empty space has come because all these 100 people moved out. Something similar is happening in plasmolysis, where all the water content has moved out, and if water is gone, you are left with very little of the cytoplasm, you are left with some cell organelles, very little of it is left. So they'll all shrink and come to one corner, and then you're left with a lot of empty space inside the cell. So this condition is what we call as plasmolysed cell, okay? All right, okay, fine. Plasmolysis is only contraction or expansion. Only contraction. I'll not even say contraction. Contraction is a wrong word. Use shrinkage. Okay. So the term that I want you to use is shrinkage. Yes? Yes, Kartika Chandra, I can see your chat. What is opposite of plasmolysis is when the cell swells up. So same cell if you put in this, no, it will become deplasmolysed. That is the opposite. Okay, it will become deplasmolysed. All right, everybody, we will move on. Yes, okay. All right, okay, everybody, very quickly. Won't the cell dehydrate? Precisely, that is what is happening, right? This is what is dehydrating. Okay, everybody, why is cell membrane also known as the plasma membrane? See, we call it as the plasma membrane because of the nature of it, okay? It's like a, see, cell membrane is of course the outermost covering of the cell, but plasma membrane, due to the nature of it, we call it as plasma membrane, okay? Yes, all right. Very good. Now moving on to nucleus, okay? So now in the case of nucleus, we know that nucleus is the control center. Now everybody focus, we are moving on to the next part, yes? So we know that nucleus is most often than not referred to as the control center, yes? So most activities that happen inside the cell, that whatever we have discussed, is regulated by the nucleus. Now we know that the nucleus has the outermost covering, which is known as the nuclear membrane, right? And this nuclear membrane has some pores in it. It's not a continuous membrane, but we see that there are some pores present, which is known as the nuclear membrane. And inside, of course, we have the nucleoplasm. But of course, inside it, we find that, yes, here's a quick recall, that nuclear membrane is a double membrane structure, has nuclear pores that allow the entry and exit of substances inside and outside the nucleus. But the main thing about the nucleus is the presence of your genetic material, right? So inside the nucleoplasm, you find the chromosomes, right? Which is nothing but the genetic material. Now, chromosomes are like rod-shaped structures, okay? So we see that they are rod-shaped structures which usually appear when the cell is about to divide or when it's about to make more of itself, right? But normally we find it as chromatin material. And at the end of the day, the genetic material that is present is nothing but DNA, right? That is deoxyribonucleic acid. And here we see that this genetic material that is there has all the information. No, let me just go back. We see that this genetic material has all the information present inside it. So right from our hair color, right from our eye color, to the fact that we should have, maybe we should carry out these activities, all this information is there in the DNA. What is DNA? Again, you will ask me, so I'm writing this down. It is deoxyribonucleic acid. Yes? So that is what DNA stands for. Now this DNA has various segments, right? So DNA is a very long structure, okay? So we know that DNA is a very long structure, and here we see that there are segments of DNA that will have this information, right? For the specific functions. Which means if the cell has to, help in movement of substances, or if the cell has to, produce energy, or if the cell has to do something small as move like this, right? I'm just giving you a random example. Cell has to do anything, all that information is present in these genes, right? So what are genes? Small segments of DNA that contain, or they are sequences that consist of information for specific functions in an organism. Now, of course, we looked at the structure of the DNA. We looked at the structure of the nucleus. We saw that there was nuclear membrane, nucleolus. We saw that there was nucleoplasm. Now, this is a very advanced version of the nucleus. We also see that there are some primitive forms, which is why, based on the type of nucleus, we have prokaryotic cells and we have eukaryotic cells. So this is based on the type of nucleus. So pro here means primitive, right? And caryon here means nucleus. So prokaryotic cells are those cells which have a primitive nucleus. That means that their genetic material is not enclosed. There is no nuclear membrane. There is no nucleoplasm. The genetic material is found concentrated in one part. See here, it is found concentrated on one part of the cell. And we call this concentrated portion as a nucleoid region. So what is a nucleoid region? A nucleoid is a region in a prokaryotic cell where the genetic material is found concentrated, right? And the most common example is that of a bacterial cell. Now, apart from this, we see that these prokaryotic cells have no true membrane bound cell organelles. So some of the advanced cell organelles like your mitochondria, your endoplasmic reticulum, you will not find in your prokaryotic cells. So this is the second point of difference, okay? This is your first point and this is your second point. Your third point will be an example. Now on the other hand, when you talk about a eukaryotic cell, U means true, not well. U means true and caryon again means nucleus. So they have a true nucleus or a well-defined nucleus and they consist of membrane-bound cell organelles. So what do I mean by a true nucleus? The genetic material is found concentrated or it is enclosed by a nuclear membrane. Are we clear? This difference between prokaryotic cell and eukaryotic cell will definitely come in your exam. Okay, this is definitely bound to come. Ma'am, chromosomes are made up of what? Chromosomes are made up of your genetic material like your DNA and your proteins. Yes, okay. Can you explain nucleoid once again? Nucleoid region is when the genetic material is found concentrated, right? So there's no nuclear membrane, okay? Only the genetic material is there. You imagine I have many ropes. Let me just show you with this. Imagine if this is my genetic material. I hope you can see this. And I'm just winding this up like this, okay? I'm making it into a bunch like this. And I'm concentrating it at one part and I'm keeping it. So here if you see there is no membrane covering it. I'm not putting it inside a case or anything. It is just concentrated here. This is what is a nucleoid region where the genetic material is found concentrated. Okay? Yes, okay, all right. Okay, you are not able to see it, is it? I am so sorry. Yes. Dia, I know you've been asking me what time class will get over. We'll try to. It's a marathon class, right? So it's 6 onwards. It will go on, okay? Yes, all right. What is RNA? RNA stands for ribonucleic acid, right? So it stands here for ribonucleic acid, and RNA here plays a very important role in your protein synthesis. Yes, so are we clear? All right. Ma'am, some line about chromatin was there. Chromatin is when the DNA or your genetic material is in thread-like form. But what happens is that closer to division, this will become even more condensed and they will form this rod-like structure. So your chromosome that is there is actually formed by this, okay? It will form this rod-like structure known as chromosomes. Yes? All right. What is ribo? Ribo is with respect to the type of carbohydrate that is there. Repeat, nucleoid. Nucleoid is when inside the cell the genetic material is found concentrated in one region. That is what we mean by nucleoid. Ma'am, समझ नहीं आया? दो तरीके का सेल होता है बेस्ड ऑन द टाइप ऑफ न्यूक्लियस. एक प्रोकैरियोटिक सेल होता है और दूसरा यूकैरियोटिक सेल. प्रोकैरियोटिक सेल में प्रॉपर मेम्ब्रेन बाउंड न्यूक्लियस नहीं है, राइट? इंस्टेड हमको क्या दिखता है वहां पे एक न्यूक्लियोइड रीजन दिखता है. वो एक स्ट्रक्चर नहीं है, वो एक रीजन होता है. और वहां पे जो जेनेटिक मटेरियल होता है ना, वो जेनेटिक मटेरियल ऐसे थ्रेड की तरह वो कॉन्सेंट्रेटेड एक रीजन में कॉन्सेंट्रेटेड होता है. इसीलिए हम वो रीजन को न्यूक्लियोइड रीजन बोलते हैं. ठीक है? दूसरी तरफ वी हैव यूकैरियोटिक सेल्स. यूकैरियोटिक सेल्स में हमारा जो जेनेटिक मटेरियल होता है, जो डीएनए होता है, वो न्यूक्लियस के अंदर और एक न्यूक्लियर मेम्ब्रेन की अंदर वो प्रेजेंट है. सो यू हैव अ वेल डिफाइंड न्यूक्लियस, राइट, विच इज व्हाई वी कॉल इट एज यूकैरियोटिक सेल. और एक और पॉइंट ऑफ डिफरेंस जो होता है यहाँ पे, प्रोकैरियोटिक सेल में हमको मेम्ब्रेन बाउंड सेल ऑर्गेनेल्स नहीं दिखता, राइट? सो यहाँ पे सिर्फ we only find ribosomes. यूकैरियोटिक सेल्स में हमको प्रॉपर डबल मेम्ब्रेन बाउंड सेल ऑर्गेनेल्स हमको दिखता है. ठीक है, अभी समझ में आया? Yes, ma'am, please. When I go slow, you will tell me, ma'am, very slow. When I'm going fast, ma'am, cheetah. Yes. Okay, why nucleus has double membrane, right? It has double membrane because of, again, most in advanced structures, we have the lipid bilayer only. So, ma'am, nucleus and nucleoid type, the type of DNA will differ. See, nucleoid in bacteria, it will be DNA, okay? But the sequence of DNA will differ. See, when I say DNA, composition, this is little high end for all of you because you're asking me, I'm telling you, okay? This is not there in your syllabus, but I'm telling you anyway. See, you have your DNA, right? This is in bacteria, this is in us, right? Now I keep drawing these lines inside, no? These lines that I draw inside are the sequences, okay? These are the sequences that I tell you. Now these will be differ from what you see in us. So although it is DNA only, it will be different in terms of sequences. Yes? Ma'am, can you repeat in easy words? Nucleus and nucleolus. Nucleus that is there. Oh, okay. The mic is at a distance. The mic is pinned here. That's why. Normally I have a mic on top, which is easy. Okay. Nucleus that is there. No, see, nucleus that is there. Nucleus is the control center of the body, right? So it regulates all the functioning. Nucleoid is a region we find in the in prokaryotic cells, right? In Hindi, न्यूक्लियस एक स्ट्रक्चर होता है जो एडवांस सेल्स, एक्चुअली यूकैरियोटिक सेल्स में हम वेल डिफाइंड न्यूक्लियस देखते हैं और वहां पे वी सी दैट देयर आर इट इज अ कंट्रोल सेंटर. वो एक कंट्रोल सेंटर और हेडक्वार्टर्स की तरह फंक्शन करता है. न्यूक्लियोइड रीजन एक प्रिमिटिव टाइप ऑफ न्यूक्लियस होता है. वहां भी वो भी एक कंट्रोल सेंटर है, लेकिन स्ट्रक्चरली जब आप देखोगे यहाँ पे स्ट्रक्चरली ये थोड़ा और एडवांस्ड होता है, ये थोड़ा सिंप्लीफाइड और प्रिमिटिव स्ट्रक्चर होगा. ठीक है? Yes, Diya, I have already answered your question. I told you this class is going to go on for a while. It's a marathon class. No? So this was all about your nucleus. Now we will move on to cytoplasm. Cytoplasm is easy peasy. Cytoplasm is the gelatinous, or it is a jelly-like, or it is a viscous fluid that fills inside the cells. It is made up of water, salts and various components which are there. We see that all your cell organelles are found in the cytoplasm, and various chemical reactions also takes place within the cytoplasm. Now where is your cytoplasm found? Your cytoplasm is found between your cell membrane and your nucleus, right? So this is all about the cytoplasm. Yes? Okay, all right, very good everybody, very good. Yes, okay. Now with this we've looked at cytoplasm. Now we're going to quickly move on to our cell organelles, okay? And then we can wind up all the important parts that are there. So and then we can wind up fundamental unit of life. Then we still have tissues and improvement, long way to go, no? So we'll go a little faster. So now we have all the important cell organelles. So we have the first cell organelle, which is the endoplasmic reticulum. Now if you see, the endoplasmic reticulum is found closely associated with your nucleus, and they are made up of many flattened, tubular structures, okay? So they are made up of many flattened tubular structures. Now what are the functions? Now first, don't go into rough and smooth. Learn about the functions of ER. What does ER do? ER serves as channels for transporting materials. This is a line just from your NCERT. Underline this. They will ask you what is the function of ER. Don't write protein synthesis, okay? Because they're not specifying RER, right? They're asking generally what does ER do. So first you should write this. What is the function of ER? They act as channels for transport. Between where to where? Between various regions of cytoplasm, or between cytoplasm and nucleus, right? What does ER also do? It also functions as a site, right? So we see that some of the biochemical activities takes place inside the ER. Now we have two kinds of ER. We have your rough ER and your smooth ER. Now we have rough ER, which is rough endoplasmic reticulum. Why is rough endoplasmic reticulum called so? Because we see that on the surface, we see that there are various structures known as ribosomes, right? So on the surface we have ribosomes. Now what does this ribosomes do? Ribosomes are responsible for synthesizing proteins. Very, very important. So they are what produces proteins, and we know proteins are the building blocks of our body. And we see that ribosomes are structures we find both in prokaryotic as well as eukaryotic cells. Now we see that these proteins are synthesized mainly by your rough ER. And we see that these proteins are then packaged in vesicles, which are nothing but small vehicles, right? So always remember, what are vesicles? Vesicles are vehicles. To do what? What do vehicles do? Vehicles help in transporting, no? So a lot of you who will ask me, ma'am, what are vesicles? They are nothing but vehicles for transport. So this is what RER does. Now you have your smooth ER. Now your smooth ER is called as smooth ER because you see there is no ribosomes present here. And we see that these are responsible for synthesizing lipids or your fats. And we know that in some cells like our liver cells, especially in some vertebrates, these are also responsible for detoxification or getting rid of toxins. What is detoxification? Getting rid of toxins, right? So that is what is going to happen. So now you have RER and smooth ER doing this. Now what happens is that your RER or rough endoplasmic reticulum is producing proteins. Your smooth ER is producing lipids. Now these are the raw materials for producing your plasma membrane, right? Now plasma membrane is the outer boundary of the cell. We find them in cell organelles also. So we see that a membrane is generated or is synthesized. This is what we call as membrane biogenesis. We know what a membrane is with respect to plasma membrane. Biogenesis. Genesis means to form, or I can say to generate. So when you're learning about your ER and you know their functions, right? We know that this is what is going to happen. Are we clear? Are we clear? Yes, I know we are still on the first chapter. We have to go faster, no? We have to be fast. Ma'am, what type of proteins does ribosome synthesize? So ribosomes, in terms of proteins, they make simple proteins which then undergo modifications, no? Yes, okay. Protoplasm is all your living components. Your cell organelles form your living components, right? So protoplasm includes all your living components of the cell, while cytoplasm includes all the jelly-like fluid that exists between your plasma membrane and cell membrane, okay? Yes, thank you for that, Himani. Thank you. Now let's quickly move on to the next one. So we are clear with RER, no? We are clear with endoplasmic reticulum. We are clear with rough endoplasmic reticulum and smooth endoplasmic reticulum. Yes? All right. Now I'm going to move on to Golgi apparatus, okay? So Golgi apparatus was a cell organelle which was discovered by Camillo Golgi. Hence the name Golgi apparatus, or sometimes you will also learn it as Golgi bodies also, right? Now Golgi bodies are again made up of flattened sacs, okay? So many flattened sac-like structures will be there. And what is the role of Golgi? Golgi is most often known as the postman, right? Golgi is our postman of the cell. So he is responsible, or Golgi is responsible for packaging, right? So it is responsible for packaging, it is responsible for modification, and it's responsible for some storage as well. But more than that, it's responsible for transporting. So a lot of the proteins which are produced by the RER will come to your Golgi, right? And in the Golgi, we see that sometimes they undergo some modifications or they get some changes so that proteins can become functional, and then this Golgi will send it away. And it will again send it away in vehicles known as vesicles, right? Which is why it is known as the postman. And of course, it helps with the formation of lysosomes. What are lysosomes? Lysosomes are another kind, right? Yes, it is another kind of, what do you say, cell organelle that we are going to be discussing. Yes, all right. Ma'am, cytoplasm is a non-cellular fluid. We'll not really say non-cellular, okay, because we find it inside the cell, so it is a cellular fluid, right? Yes, all right. Okay, everybody, all right. Ma'am, Golgi is made up of which substance? It's made up of many components put together. Substance-wise, I am not going to go in detail. Yes? Okay, all right. Suicidal bags. Yes, I will send you the PDF, not a problem. Okay. Why is ER called as the skeleton, right? So see, the main reason that is there, why ER is called as the skeleton is simply because it forms, because of the structure that it has. I told you they are tubular in nature, right? Let me just go back to that slide, right? So we see that they are tubular in nature. So they act as a skeleton for the cell, or what we call as cytoskeleton, right? And then of course we have mitochondria. We know mitochondria is a very popular cell organelle which is also known as the powerhouse of the cell, right? So we call it as the powerhouse of the cell. Why? Because mitochondria is double membrane bound. So it has an outer membrane and an inner membrane. Now this inner membrane here is responsible, or we see that it is folded into finger-like projections known as cristae. So what are cristae? Cristae are finger-like projections. And we see that they have these finger-like projections to increase the production of ATP, right? So mitochondria is called as powerhouse because it produces ATP. ATP is nothing but the energy currency of the cell, right? So we see that production of ATP takes place inside the mitochondria, specifically inside the inner membrane, okay? Yes, all right, okay. So I can see that you're asking me to explain cis phase, trans phase. Can I quickly wind up the cell organelles and then I'm going to take your doubts? Can we do that, everybody? Shall I do that first, okay? All right. Are ER and cytoskeleton the same? No. ER can form one type. It can form a basis of it, but they are not the same. Because cytoskeletal structures will have so much more, which you will learn later on and you don't need to worry, okay? Yes. Now we're going to go on to lysosomes, right? So we know that lysosomes are nothing but popularly again known as suicide bags of the cell. So why do we call them as suicide bags? Because we know that they are spherical cell organelles which have digestive hydrolytic enzymes, or we call them as digestive enzymes. And these enzymes are actually formed in the RER, okay? So RER will put these enzymes, or it will make these proteins, send it all the way to Golgi. Golgi will package these enzymes and eventually lead to the formation of lysosome. And you know that your lysosome is what is responsible for getting rid of your unwanted substances that may enter, or if the cell has been worn out and it needs to kill the cell, then it will leak its hydrolytic enzymes all throughout the cell so that the cell in itself can be gotten rid of, or it can be killed. So the main role of ribosomes, lysosomes is like your garbage disposal man. Whatever you don't need, you throw it in the garbage or you get rid of it, right? So lysosome is like a self-functioning bag like that. Then you have your vacuole. You know that vacuoles are also known as storage sacs, right? So we know that they are storage sacs. They are membrane bound structures which store a lot of water. They store a lot of other substances which we also call as cell sac, right? And we also know that it stores sugar, salt, amino acids, etc. Now in the case of animal cells, we know that vacuoles are relatively smaller, while in plant cells, vacuoles are relatively bigger because the need for storage is more in plants. Yes? Then of course we have plastids. Now plastids are exclusively found in plant cells, okay? Now plastids are cell organelles which have certain pigments in them, okay? So they have certain pigments in them. And based on plastids, we have three kinds. You have chromoplast, you have chloroplast, and then you have leucoplast. So chromoplast is when there are certain colored pigments, right? So you have some examples like you have anthocyanin. I'm just giving you an example, which is a pigment that imparts a bluish color. So you have seen some of these violet, bluish flowers, no? They will have chromoplast in them. While chloroplast we know are found in the leaves mainly. They have a green pigment in them known as chlorophyll, right? And we know that this is very, very important for photosynthesis. Then you have your leucoplast. Your leucoplast is also known as a colorless plastid, and here mainly it is required for the storage, right? So here mainly, like in the case of potatoes, you find your leucoplast. Yes? So are we clear, everybody? Are we clear? Where does the color come from? The pigment is again a chemical compound which is synthesized by the components inside the plastid, right? Yes. So this is about chloroplast. These are some differences between plant and animal cells which again can come, right? So here we know that in animal cells there are no cell wall, but in plant cells you have cell wall. In animal cells there are small vacuoles, while in plant cells you find large vacuoles. Here no plastids, plastids are present, presence of centrioles, no centrioles. Now a lot of you asked me, ma'am, you know what, how do plant cells divide if they don't have centrioles? So there are other mechanisms in which the plant cells will know that they have to divide, right? Which is why plant cells are able to divide even though they don't have centrioles, okay? So there are other mechanisms that we need to know. Ma'am, please repeat chromoplast. Chromoplast is a type of plastid that we find especially in the petals of the flowers, in the walls, in some colored fruits that are there. Whichever imparts color in plants, that's because of chromoplast. Okay? All right. Is chloroplast a kind of chromoplast? Yes, it is. It is. Okay. Now last bit is on. So with this we have covered. Now the last portion, which is a very small portion, which Ankita ma'am has already taken in detail, is cell division. Because we know from cell theory that cells come from pre-existing cells, right? So how are cells coming from pre-existing cells? By the process of cell division. So what is cell division? It is nothing but a process wherein a parent cell divides to form one or more daughter cells, right? So we see that one parent cell will undergo certain changes and give you two daughter cells. Yes? And of course, based on this, you have mitosis, which is known as the equational division, which means that at the end of the day, the number of chromosomes in parent is same as the number of chromosomes in your daughter cells, while meiosis is known as your reductional division, where the number will get reduced to half, right? Which is why we call it as reductional division. Yes? All right. Which enzyme is secreted by Golgi apparatus? Golgi produces, see, it packages proteins. It doesn't per se secrete an enzyme. Sometimes if some enzymes need to be packaged, because enzymes at the end of the day are proteins, no? They will also be sent to the Golgi, and then it will then send it to wherever the, protein needs to go. All right, everybody, all right. Okay, so now with this, I think I have wound up the first chapter. I know I took a lot of time to wind this chapter up. I will be faster. So very quickly, is the same chapter in the starting. I know it is the same chapter, but I'm done with it. So any doubts so far? Any doubts? Okay, all right. Very quickly, just give me 1 minute. My chat has got stuck. Just give me a moment, everybody. Yes, okay, all right. Cis and trans. Okay, so first and foremost, you have Golgi bodies, no? Okay, let me just explain this. See, Golgi bodies, normally what we do, no? We see that this part of it is the cis phase that is there, right? So here is the cis phase that is there. Thinner ones. But what happens is that over time, this will keep moving. See, it is a stack, right? So this will keep moving and it will move on to trans phase, okay? And once it reaches trans phase, this is where the secretory vesicles are formed, right? I'm not going into details of all of this because it's a little beyond what you know, but I hope you understand. So this is the smaller one, trans is the bigger one that is there, okay? All right. Ma'am, what is present in the intercellular space? In intercellular space, you will find extracellular fluid. Okay, so you will find extracellular fluid between two cells in our body. Okay. Yes, all right. Which cells are autonomous? Mitochondria and chloroplast are autonomous. Okay. No, Diya, I am not ignoring you. What are ribosomes? Ribosomes are membrane-bound structures which are responsible for synthesizing proteins. Yes, they are responsible for synthesizing proteins. What is the function of cytoskeleton? Providing structure. It literally acts like a skeleton to the cell. Okay, that is what do we mean by that? Okay, Mahinder, please tell me, what do you need? Yes. It is semi-autonomous. My bad. It is semi-autonomous. Yes. Okay. Meiosis, ma'am, ribosomes don't have membrane. They have a single membrane, not a double membrane. Okay. All right, ma'am, what is primary lysosome? Okay. Primary lysosome is the initial lysosome that is formed. But we normally don't say primary. We just say that we just call it as lysosomes, right? Okay, any more doubts? Centrioles function. Centrioles play a key role, right? In mitosis especially, we know that there is something known as spindle fiber formation. Okay, so when your mitosis takes place, there are various phases. Okay, and for your chromosomes to move around, right, we see that there are these spindle fibers, right? So these centrioles are responsible. It is responsible for the formation of spindle fibers. Yes. Okay, ma'am, please. All right, see, I want to be specific, right, on how, when I will finish. I will try my best to finish as soon as possible, but I'll only be able to do it with your, with your cooperation, right? So I need you to stay focused in class. All right, okay. What are chromatids? See, chromatids are these two structures you see, no? These are what you call as chromatids, right? So that's what we call as chromatids. Yes? Does vacuole provide turgidity and rigidity? Yes, it does. Okay. When will mitosis takes place? Is there a particular time or can it take time? Can it happen any time? There is a cell cycle which keeps happening within the cell, which puts the cell in check so that it happens, it's not happening uncontrollably, right? It happens in a very regulated manner. And if the cell cycle is not in check, then we see that sometimes uncontrollable growth of cells could lead to formation of tumor, right? Yes, all right, everybody. So with this, I'm going to have one more sip of water and we're going to wind to the next chapter. But more or less, are we clear, everyone? Are we clear? Yes. Meiosis is a kind of reductional division, right? So we see that meiosis is a type of reductional division that we see, which happens only in the sex cells, or more specifically, it happens in the reproductive organs, right? So we see that this happens in the reproductive organs, and mainly this is what is responsible for the formation of gametes, right? So egg cell, sperm cell, they are all formed as a result of meiosis. Yes? Okay, everybody. So now with this, I'm going to move on to the next chapter. I do understand that a lot of doubts are there, but here I'm going to, because some of it is a little out of syllabus, I will take some questions only. Endocytosis is a process wherein we see that, like lysosomes, right, they will engulf it, right? So we see that endocytosis is a process of engulfing a foreign particle, ingesting it, breaking it down within the body, and then expelling all the unwanted substances. Yes? Shall we move on to tissues? Tissues is also pretty huge, but I'm going to be very fast with tissues, okay? Shall we move on? Everybody, give me a quick thumbs up for tissues and then I'm going to start, okay? All right. Centrosomes and microtubules. See, centrosomes are structure cell organelles we find in animal cells which have your centrioles. Microtubules that are there, no, they are those cytoskeletal structures which are there. Plasmodium is a parasite causing malaria. Ma'am, which cell will divide in meiosis? You have your parental cell which is present inside the reproductive structures. There are special cells which give rise to gametes, okay? Ma'am, what is active? Active transport is when you require energy, Ria. So when energy is invested in the transport, it's called as active transport. Passive transport is when no energy is required, like in the case of your diffusion and your osmosis. Yes? Yes, please start the next chapter. I shall. Okay, so now next chapter that we're going to look at is tissues, okay? See, tissues is a very simple chapter. Tissues is also a memory-based chapter, okay? Lot of things to remember in tissues. So here's a quick mind map of what all we need to cover. See, you have tissues, you have plant and animal tissues. In plant, so many are there. In animal, even more are there, right? But what we're going to do is, whenever you are learning tissues, now I'll give you a trick, okay? In tissues, you need to know 3 things, and that's all. Make a table, learn this, it will be easy. What is the structure of the tissue? What is the function of the tissue? And what is the location of the tissue? You only need to know these things. All your questions are going to be based on this, okay? Other than that, you have nothing to worry about. Yes? But to understand tissues, you need to understand levels of organization, right? Now, this is a very simple topic and I want all of you to focus. Yes? All right, everybody, very quickly. Time is ticking. I know. See, levels of organization is when you know that in multicellular organisms, right? So, in multicellular organisms which are made up of many cells, we see that many cells which are similar in structure. Write this down, okay? So, many cells which are similar in structure and come together to form or perform, So when they come together to perform a common function, right? That is what we call as tissues. So remember, many cells which are similar in structure, right? They form your tissues and that's what we are going to learn today. But now here also we have many type of tissues, no? So all these different tissues, when different tissues come together, they form your organs. So if you take an organ like your stomach, right? We see that the stomach will be made up of few types of tissues that will be there. There will be some muscular tissue, there will be epithelial tissue, there will be, connective tissue. All these tissues are coming together and giving your flattened J-shaped structure known as your stomach. Now we have stomach, liver, intestine, pancreas, all of which are examples. Now all these different organs, they will come together to form an organ system. So if you take your stomach, your intestines, liver, pancreas, they all come together to form your digestive system, right? And we know that the digestive system here is responsible for what? It is responsible for digestion at the end of the day. So even though your stomach, your liver, all does slightly different functions, all of them together will work to form your digestive system that helps in breaking down complex substances into simple soluble forms. So you have digestive system, respiratory system, then you have, your reproductive system, so many systems in the body. And all these different organ systems will form your organism, right? So organisms like you, me, our friends, our family, we are all organisms at the end of the day, and complex organisms, because I am able to do what I'm able to do today because of all my organ systems working together. If they were not working, then I'll not be able to do my functions effectively, no? So that is what we mean by it. All right? Okay. Yes, FF8. Listen to me, ma'am. I have questions. Please tell me. Are we clear with this, everyone? I can see some of you are very distracted today in class. Why is that happening? Can you tell me? Okay, ma'am, what is, Mantesha is asking me, ma'am, what is osmosis? I will tell you what is osmosis in just a bit, right? So osmosis is nothing but a process of movement of water molecules from a region of higher concentration to a region of lower concentration across a semi-permeable membrane, okay? Yes, all right. So everything and each and everything in our body is a matter of cell 24 hours. Exactly. Very good, math and science. Very good. Is skin a tissue or an organ? Skin is an organ. It is the largest organ in our body. It's not a tissue. Okay? So now here, of course, we have the definition of tissues, and we know that group of cells which have similar shape, right, and function, or they come together to perform a specific function, is known as tissues, right? And we have two kinds. We have plant tissue as well as we have animal tissues. Now, main question is, why do we have plant and animal tissues? Why are we bifurcating the two? Because clearly plants and animals are very different from one another. Animals will move around from one place to another, while plants are fixed to one point. Plants produce their own food while animals are dependent on other organisms for food. Growth in animals will stop after a certain time, while in plants we see that it tends to grow. But again, this will also stop in some time, okay? Not that it will only keep growing forever and ever, but we see that the rate in which they grow is slightly different. And again, growth in plants are restricted. So here we see that growth is restricted to certain parts of the body, while in our case, we grow uniformly. It's not that in our case, growth is only there in the tip of our fingers, tip of our nose, or tip of our head. Nothing like that. We grow uniformly as a person. But in the case of plants, that's not the case, okay? Which is why we're going to be looking at plant tissues first, yes? So in plant tissues, you have meristematic tissue and you have permanent tissue. Now, what are meristematic tissues? So meristematic tissues are those tissues which are continuously dividing, okay? So here we see that they are continuously dividing, or they are made up of cells which are continuously dividing, and we call them as meristematic cells, right? So because they are continuously dividing or they are actively dividing, we see that they contribute to the growth of the plants, or we see that these are what are responsible for growth in plants, right? So we see that meristematic, or the cells that make up meristematic tissue, have a prominent nucleus, they have no vacuoles, they are actively dividing, they are thin-walled cells, and they have a dense cytoplasm, right? So this right here is the structure, right? What is the function? Function is growth, yes? So this is the function. And location, where do we find them? Can you tell me where will we find meristematic tissue in the chat? Can you tell me, everyone, where are we going to find meristematic tissue? Yes? Okay, all right. Very quickly, everybody, can you tell me where in plants? Don't tell me, ma'am, in plants I'll find meristematic tissue. It's a plant tissue, no? Where will we find it in plant tissue is what I'm asking. Very good. In tips of roots, leaves, buds. Simple. You can say growing regions, right? We find them in the growing regions of the plant, right? That's where we find them. Very good, everybody. Very good. Don't say different parts of the plant. If I'm asking you meristematic, you have to tell me growing, okay? Yes, very good, very good. Head region, internode, yes, wonderful, wonderful. So like based on what you are all telling me, we know that there are three kinds of meristematic tissue, right? So you learned about meristematic, now you have three kinds. What are my three kinds? Can you tell me in the chat as well? What are my three kinds of meristematic tissue? Yes? In the chat, everyone, very quickly. Yes? Apical, meristem, intercalary, and lateral. Very good. It's on the screen also for all of you to be easy. Apical, intercalary, and lateral. So again, if you see, in all this, you don't need to know structure, because see, structure-wise, they are all the same. I told you, no? We've already looked at meristematic tissue and we know what is what. But in this, we know that apical meristem. Apical comes from the word apex, right? Which means we find them in the tips. Yes? So we find them in the tips of roots and shoot of plant. What do? They help in the growth of roots and shoots of plant. They allow in the extension, right? So it's very simple, easy. Now you have lateral. So I'll do lateral first and then I'll go to intercalary. Lateral, as you know, we find them in the lateral regions, right? Or you find them in the sides of the plant, right? In the stem and in the roots, you find them in the lateral regions. And they help in the horizontal growth, okay? So your apical meristem is helping you in vertical growth. Your lateral meristem is helping you in horizontal growth, or what we say as it increases the girth, right? Girth is nothing but the width that is there. It helps in increasing the width of the plant, and it plays an important role in increasing the growth. Then you have your intercalary meristem. Now your intercalary meristem that is there is present in the nodes and the internodes. Alright, so let's focus on this. It is present in the node and the internodal region. What is a node? Node is a point from which a new branch, a new leaf, a new flower, whatever you want, new anything arises from your node, okay? So this is a node. Space between two nodes, if this is my node 1, this is my node 2, region between two nodes is what we call as an internode. So we find them in these regions, and this also helps in the elongation, right? So it helps us in the growth of the plant. Again, this would be vertical growth, right? And intercalary meristem is something we find in, some plants and not all. So like in bamboo and all, you find it, okay? So this was all about your intercalary meristem, and with this we've had a look at all our meristematic tissue. Are we clear with meristematic tissue? Super easy, no? Ma'am, is immune system tissue, organ, or organ system? It's a organ system. Because we're saying immune system, no? Whenever term system is there, you understand it's an organ system. Yes? What about lateral? Lateral helps in the lateral growth in this manner, right? What is secondary growth? So secondary growth is when the plant has matured a lot. So your meristematic tissue will be found actively growing for some period of time, okay, like in the initial stages. But what happens is that when they become a little older, right, like for example in trees and all, when they're slowly forming, we see that these meristematic tissue would have lost their ability for some time, but they'll re-get this ability and they'll exhibit a growth where these lateral meristem will start to grow on both ways. See, lateral growth is always like this. The tissue will grow always in both directions so that the width will increase. And eventually we also see secondary growth allows for formation of cork, okay? Ma'am, can you say a small definition of tissues? Tissues can be defined. Can you write this down? King of king. Tissues are nothing but they are a group of cells which are similar in structure and that come together to form a common function. Okay? Yes. Which meristematic tissue is responsible for growth? All are responsible for growth, not just apical or lateral. All of them. Okay? When will there be menti? I will wind this up very fast and then there will be menti. Okay? We'll try to wind it up by 9:30. I know it's a little late, but we'll try to wind it up by 9:30, 9:45. Okay? So now this is all about plant tissues. Now, your meristematic tissues that are there are actively dividing, right? But will these divide indefinitely? Are they going to keep dividing forever and ever? No, right? At some point they will stop. And they will stop, and what will happen to them is that they would have attained a specific structure. They would have attained a specific function, right, and a specific location, right? So they have a specific size, shape, structure. I mean, size and shape will come in structure, function, and location. And here another thing is that they will also lose their ability to divide. So after a point, they will see that they will lose their ability to divide, right? And we see that when this happens, it leads to the formation of permanent tissue, okay? We see that it leads to the formation of permanent tissue. And this process wherein meristematic tissue lose their ability to divide so that they get a specific shape, size, function, location is what we call as differentiation. Yes? So this is what we mean by it. So this process is what we call as differentiation. I hope you have all made a note of it, okay? Have we made a note? Yes, all right, very good. Manisha, I am not ignoring. I am not ignoring any of you. Which, okay, ma'am, can I write cluster of cells? You can write cluster of cells, not a problem, okay? Please repeat. See, sometimes meristematic tissues would lose their ability to divide. See, meristematic tissues are responsible for their growth, right? They are made up of actively dividing cells. But it's not like it will keep dividing and dividing forever, no? What happens is that at some point it will lose this ability to divide so that it can get a specific shape, it can get a specific size, it will have a specific function and a location, right? And this process where this conversion takes place is what you call as differentiation. Yes? Note it down. Very good, very good. So take a screenshot because I normally will not be able to send this part to you on the PDF. So you can take a screenshot, okay? Yes, all right, everybody. Nadella, I will tell you the difference between plant and animal tissue in just a bit. Okay. Ma'am, please respond to my message. Name a simple type of tissue. Your parenchyma is an example of a simple permanent tissue that is there, right? अभी हम क्या सीख रहे हैं? टिश्यू के बारे में हम सीख रहे हैं, ठीक है? Why do meristematic tissues lack a vacuole? Meristematic tissues lack a vacuole because they are constantly dividing. So they don't need anything to store food. Whatever food they are preparing, they are using it for their, they're using it in cell division, right? Which is why they don't need it. Yes, Riya, please tell me, what is it? Okay, all right. There, yes, very good. So now let's move on to permanent tissue. Now your permanent tissue are of two kinds. You have simple permanent and you have complex permanent. Simple permanent, or they get their name as simple permanent because they are made up of only one kind of cells, okay? So we see that they are made up of only one kind of cells, yes? So here we have parenchyma, which is the first kind. So here let's have a look at its structure. So we see that they are mainly made up of oval or round shaped cells, or in some cases they'll be polygonal. These cells are thin walled, which means that they don't have a very thick cell wall. They have very large vacuoles in them, okay? So we see that large vacuoles are present, and if you look at the parenchyma cells, we see that they'll be arranged something like this, okay? I'm giving you a rough arrangement. So thin cell wall, prominent vacuole, you will find a nucleus and huge vacuoles inside, right? And along with this, we see that they are relatively unspecialized. So amongst your specialized cells, they are not very highly specialized. They are very simple in that way. And we see that they have large gaps between the cells, right? So there are large gaps between your parenchyma cells. Now, where do you find your parenchyma cells? You find them in the soft parts of the plant, like tip of the leaves, you find them in the stem, right? So you find them in the soft parts which are there. And what is the main function? Main function is that they provide some temporary support, okay? Understand, I'm using the word temporary. So they provide temporary support, and they are also responsible for storing materials. Why are they storing materials? Because large vacuoles, right? So large vacuoles is a key for you to remember that it is responsible for storing materials. Yes? Now let's move on to the next one, right? Okay. Razania, again, like I'm telling you, I will tell you the difference between plant and animal tissue in just a bit, okay? Yes, all right, okay. Why are they unspecialized? See, I said they are relatively unspecialized because look at their functions. It's just storage and temporary support, right? Which is why functionally they are not doing anything way too fancy, which is why they are relatively unspecialized, okay? Yes, all right, everybody, all right. Now let's move on to some modified versions of parenchyma. So we see that in some plants, like in aquatic plants, we see that parenchyma will be modified to form aerenchyma, where we see that there will be air sacs inside the parenchyma that help these aquatic plants float. You've seen lotus and all, will be floating on the surface of the pond. They are able to do this with the help of aerenchyma. While on the other hand, in the case of chlorenchyma, we call them as chlorine because of chloroplast, right? And we know what chloroplast does. It helps in photosynthesis. Yes? Okay. Why you said temporary? Because you know that there are other simple permanent tissue which provide proper mechanical support, no? So this is like how you have a tight rope, but sometimes you'll put one tape just to be safe. Parenchyma is kind of like that. Name the tissue responsible for movement in our body. Ma'am, please tell the answer. What is responsible? Your muscles and your bones are responsible, okay? Hello, Diamond, welcome back. Okay. Yes, very good. Water lily, lotus, yes, all right. Now let's move on to the next one, which is collenchyma, okay? So collenchyma is your second type of simple permanent tissue. Now how is collenchyma different from your parenchyma? Because they are made up of elongated cells. Now parenchyma is also living cells, okay? Now collenchyma is also living cells. They have thickened cell walls and they have some irregular thickening. So if this is my cell, and this is my cell wall, we see that the corners will be thickened like this in the case of collenchyma. They will not be uniformly thickened throughout, okay? Uniformly means it should be thickened here, 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 but that is not there. It is irregularly thickened in the corners, right? Which is why we see that this is very, very important to understand. And due to these thickenings, the space is relatively less, yes? Now, where do we find them? We find collenchyma at the stalk of the leaves and below the epidermis. And it provides flexibility to these structures. So if you see, when strong winds come, leaves never fall off, right? Leaves will bend, they will go flying here and there, but they will never fall. And they're able to do this, or they have a kind of flexible motion because of the collenchyma that is present. And even in your climbers, right, in the tendrils that are there, we find collenchyma that help them with climbing up and also providing them that flexibility to twirl around, right? So that is where you find collenchyma. Yes? Are we clear? आज ही upload होंगे, don't worry. Yes. कहाँ अपलोड होता है नोट्स, कोई बताओ? टेलीग्राम चैनल पे अपलोड होता है, आप प्लीज सब्सक्राइब कीजिए टेलीग्राम चैनल पे, लिंक डिस्क्रिप्शन बॉक्स पे है, ठीक है? All right, very good. Now moving on to next one, which is sclerenchyma. Super easy sclerenchyma. Now sclerenchyma is made up of dead cells. Unlike your previous two, this is made up of dead cells. They are made up of thick lignified cell walls and they have a narrow lumen. That means that if I take my sclerenchyma, there is so much deposition of lignin in it. There is so much deposition, okay, that this space here is very less. And this space or this gap is what I will call as lumen. And where do we find them? They are dead cells. So dead cells here provide, or they are found in the hard parts of the plant, like the vein, hard seed coats, in vascular bundles, which we will just learn about, right? And their main function is to provide mechanical support. Yes? So mainly remember this, right? They will ask you these two and you need to add in the structure also. Take a quick screenshot, everybody. This is a very commonly asked question. They'll either ask you to differentiate between parenchyma and sclerenchyma. They'll ask you to differentiate between all three, or one among the three they will definitely ask. Yes? Okay. Now, very quickly, I want to UZ vlogs, do they have nucleus? Nucleus, are you asking about sclerenchyma? No, sclerenchyma will not have, okay? Answer, ma'am, you forgot my question. I didn't forget your question. I will tell you in some time. Just give me one moment. What is lignin? Lignin is a chemical substance, right? It's a chemical deposition that provides or that gives that rigidity, okay? Which tissue forms the horns of animals? It is not, it will be, what do you say, actually I don't know the answer to this. I will look it up and tell you, Himanshi. I'm not able to think of the answer right now. Okay, all right. After I finish plant tissue, I will give you the answer. Yes? What is aerenchyma? It is a modified version of parenchyma where there are air sacs, right, which are present inside it that will help, or that we find mainly in aquatic plants that help them in floating, okay? Yes, all right. Which tissue forms hair? More than tissue, I will say cells, okay? See, hair again is a part, or it's a derivative of your skin. It's not a tissue per se. They are made up of cells that are there, right? Okay. What is narrow lumen? Small space, right? This is a narrow lumen, right? No space to look through, right? It's a narrow lumen that is there. All right, okay. Now moving on to the next one, which is the protective tissue. So we have epidermis and we have cork, right? So you have leaf epidermis, root epidermis, and you have cork. Now when we talk about epidermis, epidermis is nothing but it's a set of cells which are made up of a continuous layer, okay? So they are made up of a thin sheet of continuously arranged cells, yes? And we see that there are no gaps which are present and they have thick cell wall. And we see that their primary function is to provide protection. So in the case of leaves and all, we see that the outermost covering is what you call, or what we see as the epidermis, and we call it as the leaf epidermis. So it prevents loss of water. So your epidermis will secrete like a waxy substance, okay? So it will secrete a waxy substance which will act as a coat on top of it, yes? And in the case of your desert plants and all, no, like your cactus and various other plants, this waxy coating will be super thick, right? And this waxy coating will prevent loss of water, right? So it makes sure that we know loss of water in plants takes place by the process of transpiration. And plants at all times, even though transpiration takes place, they want to minimize transpiration. They don't want to spend too much time, right? Which is why in this case we see that they have this waxy coating that is there. Apart from that, of course, it makes sure that the leaves don't get damaged easily, prevents parasite invasion, and if there is any gap, it is only for the stomata. Right? So we know that stomata are the pores which are present, so you find more on the lower surface of the leaf and relatively less on the upper side, and we see that their major function is to facilitate gaseous exchange and regulate loss of water, right? So this is what we know about stomata. Yes? Alright. Now of course we have the root epidermis. Now epidermis, again, structurally same, continuous layer. But in leaf epidermis we saw that there are various things that it does. But in root epidermis, apart from providing protection, we also see that the epidermis will form extensions known as the root hair. And this in turn will help in absorption of water. Right? So we see that it forms root hair and it increases. So this because it is single celled, right? Each root hair is just single celled. So when it is single celled, it increases surface area for water absorption, right? So in leaf epidermis, you saw that it is doing all of these functions. But in roots, functions are slightly different, right? Helping in absorption, which we don't really see in leaves, right? Absorption of water by roots. Yes, very good, very good. Now we have cork that is there. So we know cork is found in woody stem of old plants, right? So formed, again, this is made up by, as a result of secondary growth, cork formation takes place. We know that cork here is made up of dead cells, right? So we know that it's made up of dead cells. It has depositions of something known as suberin in it, right? And again, functions are the same. It minimizes water loss, prevents mechanical injury. Okay? Are we clear, everybody? Are we clear? I know, Arpan, I know. I hope all of you have had dinner. If you've not had dinner, just get your dinner as you are watching the session and you eat it. Okay? Yes, all right. Ma'am, are tissues present in the brain? Yes, tissues are present in the brain. Ma'am, explain cork once again, please. Cork is very simple. Cork is formed, or it is formed as a result of dead tissues, okay? So we know that in older plants, secondary growth takes place and it will grow on both the sides, increasing the girth, no? So what happens is that your epidermis that was initially there in the stem and in your leaves, so epidermis is also there in your stem, right? So the epidermis that is there in your stem will now get pushed out, okay? And they'll get disrupted, eventually leading to the formation of cork. Now cork is again made up of dead cells, right? And we see that these cork that is there, main function is to have or reduce water loss. So if any loss of water takes place, it will not allow water to go. And again, of course, it will prevent the inner parts from getting damaged, right? So that is also there. And of course, we know that it has depositions of suberin in it, which is what gives it these properties. Okay, all right. What is stomata? Stomata is nothing but pores which are present on the underside of the leaf, right? And it facilitates gaseous exchange. So carbon dioxide from outside will enter, oxygen will go out, all of this gaseous exchanges takes place. Is it also known as secondary growth? Yes, it is. It is known as secondary growth. Okay, so here's a quick recall of your leaf, root and cork cell. Are we clear, everybody? Are we clear? What is tensile strength? So if you're stretching it, no, like for example, if I tensile strength of a rope, imagine how much of it can it withstand before it actually tears off. That's what we call as tensile strength. Yes? If branches of leaves if branches and leaves fall due to strong wind, does it mean collenchyma is absent? No. See, there's a level up to which your collenchyma can provide flexibility and support, right? It means that the wind was that strong that the collenchyma could no longer withstand that kind of force and pressure, which is why it fell down, right? Not because of anything else. Okay. All right, everyone, can you explain root epidermis once again? Root epidermis is when the epidermis will form root hair, okay? They are single cell structures, and we see that this helps because they are single cell, no? They'll be like this, okay? It is a single cell that is there. It will help in the absorption of water. Okay, now with this we've looked at simple permanent tissue and protective tissue. Very quickly moving on to complex permanent tissue in plants, right? So why do we call it as complex permanent? Because they are made up of more than one type of cells, right? And we know that this mainly includes your xylem and phloem. And your complex permanent tissue are also responsible for transport, right? So they are responsible for transporting some important substances, yes? So in this case, we see that you have xylem and you have phloem. Now your xylem is mainly responsible for transporting what? It is responsible for transporting water and minerals. From where to where? From the soil, or I will say from the roots to the upper parts. And here we see that transport is unidirectional, right? So transport is unidirectional. Now, what are the components of xylem? You have tracheids, vessels, parenchyma, and fibers. Yes? So, what are tracheids? Tracheids are long elongated cells. Okay? They are dead cells. They help in transport of the water. Then you have vessels, which are made up of many cells which are arranged one after the other. But what happened was when they got arranged, these walls disappeared between them so that they could form one continuous tube. Now your tracheids and vessels are what is responsible for transporting water and minerals. Now you have xylem parenchyma, which is responsible for storing food materials, and you have fibers which provide support. Now in xylem, tracheids, vessels and fibers are all dead, okay? They are all dead cells, while only your parenchyma is made up of living cells, right? Okay, so very, very important that you understand. What are the components of xylem? Tracheids, vessels, xylem parenchyma, and your fibers that are there. Tracheids, vessels, and your xylem fibers are made up of dead cells, while your parenchyma here is made up of living cells, right? Now these two are what help you in transport. This helps you in providing mechanical support. Parenchyma is providing storage. Easy peasy lemon squeezy, right? Now you have your phloem. Phloem here is responsible for transporting, or this is responsible for transporting food materials. From where to where? From your leaf to upper parts as well as your lower parts. So here we see that the transport is bidirectional, okay? It's going both up and down. What is there in your phloem components? You have 5 things here actually. Oh, 4 things. You have sieve tube, you have sieve cells, companion cell, parenchyma, and fibers. So your sieve tube are these elongated structures that you see, and in between that we see that there is a sieve plate also. Now we see that companion cell is what is closely associated with the sieve tube, and they control the activity of the sieve tube. Then you have phloem parenchyma, again whose main role is storage, and here phloem fibers which are made up of sclerenchyma that provide support. So here your sieve tube, companion cell, parenchyma are all living cells, while this is made up of dead cells. Yes? All right, okay. So very quickly we have a comparison between xylem and phloem. So of course it is hidden, but you know that the components of xylem include tracheid, vessels, it includes your parenchyma and fiber. Here sieve tube, companion cell, parenchyma, fiber. So here main thing to remember is difference between xylem and phloem, okay, because they keep asking you this. What is the difference between function of xylem and phloem? Very important question. Point number 1. Transport water and minerals. Transport is unidirectional. Okay. While what about in this case? It helps in transporting food, and transport here is bidirectional. So are we clear with xylem and phloem, everybody? Very quickly. Are we clear? I like how Toxic Boy is giving us some match updates as well. Yes, we do have Menti. I am trying to wrap up the session as soon as possible, right? So let's quickly wind up the session and we'll go to Menti, okay? All right. Yes, I think my phone has gotten stuck. Just give me one moment, everybody. My chat is loading. Okay, all right, everybody. So I think my phone is stuck. So just give me one moment. Yes? Okay, everyone, so since my phone is stuck a little, I may not be able to see your comments. I'm just going to go to the system there and check your comments. We will quickly move on to the next set of topics because I'm going to check your doubts from there, okay? Because I'm not able to see it. Okay, all right, everybody. So now we're going to move quickly. In the next half an hour, I'm going to aim to complete the remaining part of it, okay? So now, of course, after this, so till my chat loads, I'm going to go on to the next part. So some of your doubts that you have right now, what I will do is when my chat comes back, I will be able to answer it, okay? So till then, I want you all to focus. We're going to move on to animal tissues. And see, animal tissues is a very big part, but it's also super duper easy, okay? There's nothing much in animal tissues. Again, structure, function, location. I'll take your doubts towards the end, yes? So in animal tissues, we have epithelial tissue, muscular tissue, connective tissue, and nervous tissue, yes? So your epithelial tissue that is there is what provides protection, right? So we see that this is what provides protection in the body. You have your muscular tissue, which is responsible for movement, right? And last but not the least, you have connective tissue, which is responsible for connecting and binding different organs together. And you have nervous tissue here, which is responsible for relaying or transmitting information, right? And I know that Razangna had a question on, ma'am, what is the difference between plant and animal tissue? See, plant tissues that are there are made up of different kinds. They're made up of plant cells, right? And we see that the level of organization we observe in plants and animals are different. So plant tissues are specialized for plants who cannot move around, who require storage, and whose growth is not uniform. While animal tissues are specialized in a way where growth is uniform, where we are able to, move around, and of course we know that storage of course is not that much of a requirement in the case of animals. So this is the main point of difference between plant and animal tissue. I hope that I have answered your question. Yes? So now quickly let's first move on to epithelial tissue, right? So let's understand what do we mean by epithelial tissue. Yes? Are you all able to see my screen? Video is struggling, video is lagging. Yes, Ashish, I'll be fast, don't worry. Okay, all right. So I'm going to be faster, right? So now, of course, in epithelial tissue, we see that this usually forms the outermost covering of organs. So we see that it forms the outermost covering of most organs in the body. And of course, it forms the inner lining, right? It forms the inner lining of cavities. So inner lining of your blood vessels, inner lining of organs, all of it is where you find your epithelial tissue. Yes? Now, of course, in your epithelial tissue, we know that there are various kinds of epithelial tissue. You have squamous, cuboidal, stratified, and columnar. Now, one thing you need to understand about epithelial tissue is that apart from protection, right? See, it forms the outer covering of your cells, right? So, it forms the outer covering of organs, so it provides protection. But it is also forming the inner lining of the organs. So they also facilitate, or they are involved in some transportation as well, or they regulate it. So it's important you understand this particular point, okay? Now apart from this, we have squamous epithelium. Now your squamous epithelium that is there comes, or the term squamous epithelium comes from the term squama, which means flat. So cells that make up your squamous epithelium are made up of flattened cells like this, okay? So they are made up of many flattened cells, yes? And we see that they have a prominent nucleus. And we know that these squamous epithelial cells are found resting on something known as a basement membrane. Now your basement membrane that is there is a structure that separates, right? So we see that this separates your epithelial cells. So if these are my epithelial cells and this is my basement membrane, which I'm going to highlight like this, it separates my epithelial cells from my underlying tissue. So whatever tissues are present, my basement membrane will separate these two, right? So squamous epithelium is made up of flattened cells, right? So acts as a protective layer, prevents mechanical injury, drying up, and of course allows diffusion to occur. So where do we find our squamous epithelium? Write down 3 places where you will find it. You just need to know 2 to 3 examples in terms of location. Don't try to mug up whatever is there in the textbook. Remember 2 to 3, it will be more than enough, right? So what all do you need to know? We find it in the inner lining of the alveoli, right? So we find it in the inner lining of the alveoli. We find it in the inner lining of blood vessels, right? Of blood vessels. And we find it as the outer protective layer. But like I told you, 2 are enough. Don't need to go overboard and learn 4, 5. Next, you have cuboidal epithelium. Cuboidal epithelium are made up of cube-shaped cells which have a prominent nucleus in the center. Here cells are flat, here cells are cube-like, right? And here we see that it mainly helps, apart from protection, I'm telling you, okay? All of it is protective, but apart from protective, this helps in secretion and absorption. Now, where do we find it? We find it in the inner lining of the kidney tubules that are there, right? So, we find them in the inner linings of kidney tubules. Now, next up, what do we have? Next up, we have stratified epithelium. So, stratified comes from the term strata, right? So, strata means layers, right? So, they are made up of many layers. Or we see that cells are stacked up one after the other, forming layers. Where do we find this? We find this in our skin, right? So, skin is made up of many layers of your squamous epithelial cells. So, their main function is to provide protection. Then you have columnar epithelium which are made up of pillar-shaped cells like this, okay? So they are made up of many pillar-shaped cells where the nucleus is found at the base. Here nucleus is in the center, here nucleus is in the base. Where do we find them? We find them in the inner lining of our intestines, right? So we find them in the inner lining of our intestines. And what does our intestine do? Our intestine helps in increasing or allows absorption. So I always tell you, if you know structure and if you know function, location is very easy. If you know that in the inner lining you will find this, then you know it's very obvious, no, where else it will be there, right? So don't worry. So for those of you who are saying, ma'am, points are not clear, please try to change the settings and you will be able to see it, okay? Yes, all right. Just refresh your page or don't worry, Telegram pe to I will send this, so not a problem. So this is all about your 4 types based on their structure: squamous, cuboidal, stratified, columnar. Now you have modified versions of it, okay? So these are some modified versions of your columnar epithelium, or I'll say just modified versions of your epithelium. So sometimes what happens is that there could be some finger-like or there could be cytoplasmic projections, okay? What am I finding? I am finding cytoplasmic projections, right? So these cytoplasmic projections will appear hair-like, okay? And we call these as cilia, okay? I'm going to write it outside. We call this as cilia. Now these, if an epithelial tissue have cilia in them, we call them as ciliated epithelium. So they bear, on the free ends, they bear cilia, and these cilia will lash back and forth in a rhythmic movement. Like, you know how sometimes in the auditorium or in concerts, everybody will be waving their hands together like this, no? You can just imagine your cilia waving hands like that, okay? So they wave, or imagine it to be something like that. And of course, that's what we mean by rhythmic movement. So they lash back and forth, thereby helping in movement of substances. Where do we find them? Find them in the inner lining of the trachea, the oviduct, where we see substances moving, right? Or we require substances to be trapped. So this is about ciliated epithelium. Then you have glandular epithelium. Glandular epithelium is what forms your glands. So what are glands? They are nothing but a group of specialized cells, or they are organs which have the ability to secrete certain chemical substances, right? So we see that they secrete certain specific substances, and we find them in the inner lining of stomach, intestines, we find them in the salivary glands, right? And we see that normally they will form a duct like this or a tube like this. Not all the time, but sometimes, right? So we see that they will form something like this. Here all the secretions will be there, and the secretions will be there, and then it will be transported out. So you find them in the lining of stomach, intestine, and you will also find them where? You will also find them in salivary glands, right? So this was all about it, yes? Okay. So now, of course, after this we have muscular tissue, right? So in the case of muscular tissue, we have, so in the case of muscular tissues, right, we know that muscular tissues are responsible for movement. I can see some conversation on the chat, okay? Can you all pay attention, please? Yes, okay. Ma'am, are endocrine glands and exocrine glands the same? No, they are not the same, okay? Endocrine glands are those glands, okay, they will, you know, they'll have certain specialized cells which will secrete your substances. These which form a duct, no, be like, for example, in salivary glands, right? They will form a duct and then they will transport it out. They are not the same. Yes? All right, okay. Now moving on very quickly to muscular tissue. I'm going to wind this up real quickly, okay? I'm not going to spend too much time. Improvement also, I've recently taken a one shot, so I'm going to do a quick revision only. Don't worry. Lot of you are getting scared saying, oh my god, this ma'am is going to take forever to teach improvement also. Are we going to really finish this today, or it'll go till tomorrow morning? Don't worry. Improvement, I'm going to give you quick revision only because I've only taken your one shot, right? And of course, we've had it very recently, more importantly, because this was your midterm syllabus, no? That's why it's taking some time and you have doubts. Improvement will be very fast. Don't get worried. We'll wind up soon. So let's come back to your muscular tissue. So muscular tissue is responsible for movement. And muscular tissue are made up of muscle cells, right? And these muscle cells have specialized structures, or they have certain proteins known as contractile proteins, right? So we know that there are contractile proteins which are present in them, and muscular tissue or muscles will normally contract, they will contract and they will relax. And that is how we are able to move. Now again, we have three kinds of muscular tissue. We have striated muscles, unstriated muscles, and we have cardiac muscles. Now your striated muscles are those muscles which are long, cylindrical and unbranched. Please write these points. They're very important, okay? And we see that in these cells, no? We see that the cells will have alternative dark and light banding. Okay, so we see that there are alternative dark and light bands, yes, which is why the term striated. Now we see that these muscles are found attached to our bones, which is why they are also known as skeletal muscles, right? And we see that skeletal muscles or movement in our hands and legs are something which we can control. Hence we call them as voluntary muscles, right? And they help us with voluntary movement, like movement in our arms, our neck and our feet. And these muscles make up about 40 40 to 50% of the body, right? So this is what we mean by striated muscles. Now we have unstriated muscles. We also call them as smooth muscles because unlike our striated muscles, they have no alternative dark and light bands, right? These are uninucleated, while in this case they were multinucleated. These cells are spindle shaped, right? While in this case they are long and cylindrical, right? And these are spindle shaped, which means they will look something like this. Yes, and we know that they are found in the walls of organs and blood vessels. So like in the inner linings of your stomach, your intestines, your blood vessels, we find the smooth muscles. And we cannot control their movement, no, whether it's your intestine or stomach or whether it's your blood vessel. You cannot control the movement, which is why they are responsible for involuntary movement. Involuntary is that which we cannot control, right? So we call them as unstriated or involuntary muscles. And last but not the least, we have cardiac muscles, which are also known as heart muscles. These muscle cells are branched, okay? They look a little bit like your skeletal muscles, but they are not, all right? So in skeletal muscles, we find that they are long and cylindrical, but here we see that they are cylindrical, but they are branched. And they are uninucleated. And apart from that, we also see that they have some structure known as intercalated disc that helps with the rhythmic movement of the heart. So where do we find it? We help in it's found in the heart, responsible for the rhythmic movement of your heart. Okay? So with this, of course, we have covered muscular tissue. So far, any doubts? Yes? Okay, all right, yes. Ma'am, are these branched or unbranched? Yes? Are we, heart muscles are branched. Skeletal muscles are unbranched. Yes? Okay. How much time for Menti? So for those of you who are asking, and I know some of you are feeling extremely exhausted also because we've been having this class, but due to the fact that the previous session had to spill over, we couldn't really start at 6 p.m. No, we had to start at 6:40. We lost almost 40 minutes. So what we can do is we will try to wind it up as soon as possible, but I need your cooperation. In the chat, if you keep asking me, ma'am, when Menti, then you are not focusing, no? That means that I'm going to have more doubts, and then I'll end up spending a lot of time explaining your doubts. So I don't want to do that. I'll be as quick as possible. I'm being as brief as possible with the explanation. I need your time and focus so that we can wind up as soon as possible. Yes? Okay, all right. Are involuntary muscles branched or unbranched? They are unbranched. Yes? Okay, all right. Very good. Very quickly, everybody, can you tell me a quick excitement level check, a quick Josh level check, so that I know that the energy that I'm giving out to my students is the energy that you are giving me, and you are still, excited for what you are learning, and you're not like, "Arey ma'am, abhi padhna hai kya? Abhi 9:30 ho gaya," right? Very quickly, I need you to show me an excitement level check because a lot of Menti request is there, right? But very important, know that you have to learn. That's what we need to know. Yes? Josh not down. Then I am happy. Okay, very good. All right, so my phone is stuck again, so just give me a moment. All right, everybody, let me have a quick check. All right, everyone, so my internet is down a little on my phone, right? So just give me one moment. Okay, so in the meanwhile, till my internet loads, I'm going to go on with connective tissues because my phone is stuck again. So we'll move on to connective tissues, okay? So connective tissues are those tissues which are responsible for connecting and they are responsible for binding different parts of the body together, and they provide a framework. Now, connective tissue, most often than not, you guys tend to get scared because there's a lot of information on it, right? So there's a lot of information, and that's why we tend to get very worried on, ma'am, connective tissue, so much to remember, what do we do, right? So you tend to get worried at this part, which is why I'm going to make it very easy. Now, in the case of connective tissue, right, we know that connective tissue here has something. So this is what I want you to focus. So in connective tissue, there are cells which make up the connective tissue that are found in something known as a matrix. Okay, what is a matrix? Not the movie Matrix, but it's a substance where we find the cells. Okay, now this matrix can either be, it can have some fibers in it. Okay, this matrix can either have some depositions in it, or it can be dense, right? Or this matrix can be fluid, right? Which is why in this case, what do we observe? We see that there are three. So based on the kind of matrix that is there, right, we observe that there is loose connective tissue, dense connective tissue, and you have fluid connective tissue. Yes, easy peasy. Okay, so let me have a quick look at the chat. Okay, all right, everybody. So since my internet is down, I'm just going to get it recharged maybe, okay, because my internet is also down with all the teaching that is happening. So I'm not able to see your chats. So just give me one moment. Okay, so I'm going to go ahead, everybody. So now, of course, as we know, right, so you have three kinds of connective tissue, and this is how we define it. It connects and binds different parts of the body, provides framework, and kept at a proper place. Now you have, first we will look at loose connective tissue. Now I told you that in loose connective tissue, we know that it has some fibers. So your matrix has some fibers, and the cells are randomly scattered, okay. So cells are very loosely packed, hence the name loose connective tissue. Now you have areolar connective tissue. Now areolar tissue, you can always think of bubble wrap, no? So when you order from maybe some of these online shopping websites, or from maybe when you do online deliveries like from Amazon and everything, you know that all your packages come in bubble wrap, no? And why do they come in bubble wrap? So that they can be packaged, so that nothing gets damaged. So your areolar connective tissue, just remember the function, provides support to internal organs, okay? So they provide support to internal organs, they fill up the space inside the organs. So they act as a packaging tissue that is there. And of course, apart from that, it also helps in repairing the tissues and also helps in phagocytosis. It also gets rid of some of the foreign particles that are there. Yes? Then you have adipose tissue, right? So adipose tissue is a kind of tissue that is responsible for storing reserve fat, right? So adipose tissue has certain cells in them known as adipocytes, which are responsible for storing fat, no? And here we see that they act as heat insulators, or they act as shock absorbers, and they act as padding around most of the organs like your kidney, your skin, right? So they act as padding amongst most of the things. So your loose connective tissue has cells which are loosely scattered. Your extracellular matrix that I told you has some fibers in it. Both are acting as packaging material. Your areolar is like your bubble wrap, and this is like your insulator that is there. Very simple and easy. Now moving on to dense connective tissue. In dense connective tissue, we see that the cells will have some depositions, or they will have fibers, right? So if they have depositions in them, we know that there'll be bones and cartilage. If fibers are there, there'll be tendons and ligaments. Now in the case of bones, we see that bones are made up of. So some of you had a question, ma'am, bones are living or dead? See, bones are made up of living cells, so they start off with living cells known as osteoblasts, okay? But these osteoblasts will become osteocytes eventually, right? Which may be like old, worn out and more matured bone cells, right? And we find these bone cells which are embedded in a matrix or their surrounding, which has a lot of calcium and phosphorus that gives the bones rigidity. And what is the function of the bones? Bones are rigid in nature. It's part of the skeletal system, right? And of course, it anchors the muscles, protects the internal organs, helps in locomotion, and so on, right? Okay, all right. So now, of course, let's quickly have a look. All right, everybody. So now we will quickly move on to cartilage, right? So in the case of cartilage, we see that unlike bones, they are made up of widely spaced out cells, okay? And we see that inside the matrix, instead of having some hard calcium, phosphorus, and all of that, they have proteins and sugar. And we see that the main function of the cartilage is to provide flexibility. So we find cartilage where all? In the tips of our nose, we find them in the tips of our ears, right? And it smoothens the bone surface. So between two bones, right? Between two bones also we will find cartilage. So that, bones being as hard as they are, if they keep rubbing against each other, there'll be a lot of friction, right? Which is why we find them between the two. Then of course you have tendons and ligaments. You know that tendons, there's a very easy way in which I remember this, okay? So you have BTM and you have BLB, yes? So what is BTM? Tendons are fibrous dense connective tissue which connect bone to muscle, okay? What is ligament? Ligament are fibrous structures which connect bone to bone. Very simple and easy. Remember your BTM and BLB, you will be sorted out. Now in the case of ligaments, we know that they are a little more sturdy because they're connecting two bones. It needs to be pretty strong, right? While on the other hand, in the case of, your tendons need to be more sturdy because they're connecting muscle to bones, while your ligaments are a little more elastic because they're connecting two bones together. And then last but not the least, we have fluid connective tissue. So we know that fluid is when the extracellular matrix is fluid in nature, right? And here we see that the functions are mainly transport of gases. And when we talk about fluid connective tissue, it includes your blood and it includes your lymph, right? So in the case of blood, which is what is there in your syllabus, we know that there is a fluid component or the fluid matrix which is known as plasma. You have the cellular components which include your RBC, WBC and platelets, right? And their main function is for transport of gases, nutrients, elimination of waste, and of course you know that your RBCs are responsible for transporting oxygen, WBCs are responsible for fighting off unwanted substances, platelets here are responsible for clotting. So with this, if you see, we have quickly covered connective tissue. I'm going to do nervous tissue as well and we will wind up the chapter, right? So nervous tissue is the one that makes up your brain, right? Or it makes up your nervous system. And a lot of you are asking me, ma'am, what tissue makes up the brain? That is nothing but your nervous tissue. Now your nervous tissue or your nervous system is responsible for coordinating various activities that are taking place within our body, right? So whatever keeps happening, it is controlled and coordinated by your nervous system. Now, of course, if you look at all the components of the nervous system, it includes your brain, it includes spinal cord, right? And of course, it has nerves. But the fundamental unit of all of it is a neuron, right? So what is a neuron? It is a basic structural and functional unit of a nervous tissue, right? So we see that it has a cell body, right? Then we see that it has structures known as dendrites. Yes, it has a long elongated structure known as axon. Then this axon is surrounded by a myelinated sheath, right, or a myelin sheath. Then of course it ends in axon terminals. No? Now we know that the cell body is the main structure and from that we have cytoplasmic projections known as dendrites. Now these are like branches of a tree, okay? They will branch out. And one long branch that acts as a trunk is what we call as the axon. And this axon is surrounded by myelin sheath which ends in axon terminals. Yes? Now we see that I told you that it is responsible for controlling and coordinating various activities. So how is it controlling and coordinating? So we see that it does this by transmitting some electric signals, right? So they talk to each other or they communicate to each other by using electric signals, right? So these electric signals will be first received, right? So we see that these electric signals will be received by the dendrite and it will go all the way, it will be transmitted here and then it will be passed on to the next neuron, right? And we see that so these axons that are there, no? These axons will get bundled up in a myelin sheath forming a nerve. So what is nerve tissue per se? When all the axons are bundled together forming your nerve fiber or your nerve tissue is what we call as a nerve, right? So this was all about your animal tissue. Are we clear, everyone? Yes, let me quickly have a look at the chat. Okay, all right, everybody, are we clear? Understood? How many chapters pending? Next chapter you see how we'll finish fast, don't worry, okay? Ma'am, synapse, yes? So synapse is nothing but a small gap that is present between, so if you see between two neurons, right, they are not attached to one another, but there is a small gap which is present between the nerve ending or the axon terminal of one neuron and the dendrite of the other. So we call this as synapse, yes? Cell body is also known as cyton. Very good. So synapse is the small gap that is present between the two, okay? Yes. Okay, everybody. So with this we come to the end of tissues, and now quickly we will do improvement, okay? Menti, I know everybody is dying. I'm waiting for Menti. So can I promise you all one thing? If you give me 15 minutes, I'll wind up improvement in food resources. Will you wait for 15 more minutes? Yes, 15 more minutes and I'm going to wind this up, okay? What are glial cells? Glial cells are these cells which are attached to your neurons, which act as connectors of different neurons, okay? Yes, you will give me 15 minutes. Quick Josh check also. Yes, okay, all right. Thank you so much, everybody. You are being such amazing students. 50 minutes. Nervous tissue can control other tissues. Yes, the functioning of other tissues it can control. Okay? Okay, all right. Till 10 p.m. we'll be studying. It's a marathon session, right? So I'm sure you've already attended Saurabh sir's marathon as well. So of course, you know, in this also some of the marathon sessions will get stretched up, and I already told you we started 40 minutes late. So yes, हिंदी मीडियम को भी पढ़ाते हैं बायजूस में. हम हिंदी में ट्राई करते हैं पढ़ाने में, but mostly मैं इंग्लिश में पढ़ाती हूँ, तो ट्राई करती हूँ, ठीक है? All right, ma'am, what is node of Ranvier? Node of Ranvier is the point, okay? So when these myelinated sheets are there, there are points at which we have the node of Ranvier. That is what we mean by it. Okay, I asked you for 15 minutes. You told me, ma'am, yes. So I'm going to start with improvement in food resources. So we know that the food that we eat is mainly of two kinds, right? So I'm not going to be looking at the chat. So I want you to focus. And then only I'm giving you Menti. Next 15 minutes, if you don't focus, then Menti will not be there. This will go on for another half an hour. You don't want that, no? But see, in all fairness, I will give you the best content in under 15 minutes, right? But if you want that, if you want a quick revision, I need your focus, right? How you have been focusing so far, I will want that again, okay? So very quickly, we are going to be looking at improvement in food resources chapter. To be honest, this is a very easy chapter, okay? Just again, when we are looking at this chapter, you need to understand some basic points. Now, when we talk about improvement of food resources for plant-based products, it will take you more time than animal-based. So accordingly, we will go, okay? So we have sources of food that we eat, and we know food is necessary because food is the fuel that drives our body. And you have plant-based food items and you have animal-based food items. Now, over time, there has been an increase in the demand for the food that we eat. And of course, to meet the growing demand, people started to over-exploit the natural resources that we had, right? Which is why there was also a need for some sustainable practices, yes? So they needed some sustainable practices, and they also wanted to make sure that whatever techniques we do to improve our food resources, they should be accessible to people. People should be able to buy it. Not like I spend 10,000 rupees in just growing one set of land and, you know, I am selling it at 20,000 rupees. No, doesn't make sense. I must be able to sell it at a price where my people, the people I am selling to are able to buy it, right? So which is why we see that there is a drastic need to improve food resources. Now when we talk about plants, we know that plants have different conditions for growth, different soil types, different light, different geographical conditions, and based on that we have our three kinds. We have kharif crops, rabi crops, and we have zaid crops, right? But keeping all of this in mind, people have devised techniques wherein they have been able to improve these food resources, right? So how can we do it for plants? We can do it by improving the crop variety. You can improve the crop variety, whereby you will have a better selection of seeds. Now once you have done that, you can improve your crop production management, so that after getting good quality seeds, you are still managing with whatever money that you have, your practices are proper, and crop protection. So once your seeds are ready, your practices are ready, you just need to make sure that you protect it. So how can we improve our crop variety? Now, the reason behind why we need to improve our crop variety is simply because some of the plants, right? Farmers were also struggling. We saw that there were some crops growing with very low nutritional value, some crops were malnourished, some pests and insects were coming and attacking the crops, low quality seeds. Farmers were in a real fix, right? Which is why scientists were like, hey, okay, we should come up with something where the seeds are only really good, so that they will not struggle with all of these problems. So the main objectives of having crop production improvement, right, or crop variety improvement, is to improve higher yield, so that in a given piece of land, right, so in a given area, more amount of crops are being able to grow. What is yield? Yield is amount of crops that are harvested per unit area, right, or per acre, sorry. Then of course you want improved quality. If I am, making my own crops, if I'm improving my variety of crops, they should be of high quality. So if I am telling that my proteins or these pulses are very high protein, they should have protein. No? Then they must be able to resist biotic and abiotic resistance, right? So all these pests, insects, they must be having the ability to resist it, right? And in the case of abiotic conditions, like for example drought, or in the case of extreme conditions, they must be able to resist it. And of course with that comes wider adaptability, desirable agronomic character. So if I want to feed my fodder, right, I must be able to see this, right? Yes, okay, all right. Now everybody, I can see some of you are getting distracted in the chat. I need your focus here, okay? You promised me 15 minutes, so you should be focusing. Now of course there is change in maturation duration as well, right? So we must be also able to grow plants which have lower maturity time. So how can we achieve it? Hybridization and genetic modification. What is hybridization? Very, very important, crucial, right? So hybridization is the crossing of genetically dissimilar plants, which is your keyword. You will get this in your exam, so please focus everyone, right? Yes, okay. So here we know that when we cross genetically dissimilar plants, we get, this process is known as hybridization. So if I have a plant which has drought tolerance, another one with large seeds, I will cross it and get drought tolerant as well as large seeds, right? Now this hybridization can happen in three ways. It can be intervarietal, interspecific, and intergeneric. Intervarietal is when we cross two different varieties, example being Kalyan Sona. Interspecific is when we cross two species of the same genus, like your Tangelo. And you have intergeneric when we cross two different genuses, right? Like your cabbage, you have radish and cabbage. So although hybridization is giving you an improved quality, we see that it's taking a long time for us to get results, which is why genetic modification is something that is required. So genetic modification is when you add a desirable gene to a genome of a crop, and then you modify the genetic material basically. So you modify it so that you get something that you want, and that's what we call as genetic modification. And this organism is a genetically modified organism. So you have BT brinjal, BT cotton, golden rice, all of which are your examples. So with this we've seen crop variety improvement. Now we have crop production management, right? So in production management, we need to make sure that our nutrients are managed properly, our water resources are managed properly, and our cropping techniques are proper, right? So in this case, in the case of nutrients, we know that most likely soil has nutrients. It has important macronutrients and micronutrients. But when we are looking at crop production at such a large scale, no, it's very difficult to keep track of how many nutrients are there in the soil. And most often than not, these nutrients can get depleted also, which is why we need to add nutrients. So we can add it in the form of manure or fertilizers, right? So manures are organic matter which are derived from soil and animal waste, right? So of course some benefits of manure is that it has increased holding capacity, makes the soil porous, increases friendly microbes and improves soil texture. But the disadvantage being that manure being an organic and eco-friendly thing, we also know that it is slightly difficult to measure how much of nutrients you have, which is where your fertilizers come into the picture. So fertilizers are inorganic chemical substances, right, which are manufactured in factories. Short term, they have good benefits like fertility of soil, portable and easy, they are quick solutions. But long-term benefits are, of course, they cause water pollution, injurious to health, and of course, we see that they reduce soil fertility, right? Yes, all right, okay. मैम बट बायो में फूड रिसोर्सेज नहीं आ रहा है, बट लेकिन ये एक कंप्लीट फाउंडेशन फेस्ट है जहां पे हम आपका सारा सिलेबस रिवाइज करेंगे, इसीलिए हम इसमें फोकस करेंगे, ठीक है? ऑल राइट, आप सभी यहां पे फोकस्ड है? यस, ओके, वेरी गुड एवरीबॉडी, वेरी गुड. आई नो यू आर वेटिंग, गिव मी 10 more minutes, it will be done, right? So this is about manure and fertilizer in nutrient management. But the thing is, both manure and fertilizers, if you see, manure has more advantages than your fertilizers, right? Which is why fertilizers are not something, although they have short-term benefits, right? That they enhance the fertility of the soil and of course you tend to grow crops. But the main point, or I would say the problem here, is that overusage of fertilizers is not recommended. Which is why organic farming, where you use more amount of manure or eco-friendly products, is what is recommended. So you use minimal use of chemicals, you use organic matter, recycled farm waste, right? And you practice proper cropping systems, right? So that you will be able to grow it better. So this is all about your nutrient management, okay? So in organic farming, you should know these things, that you are using eco-friendly products, you are not using much chemicals. These are two main points you need to know. Now next up we have irrigation. Irrigation, we know, is the providing of water at regular intervals. And in India, who is an agricultural country, we are mainly dependent on rain, right? And if there's not enough rain, or if in some cases what happens is that if there is more of rain, then we know that in both cases it's a problem, right? And drought can also affect the crops. Which is why irrigation methods include, you know, like your canals, your ponds, tube wells, right? And then of course we have all of this as well. So you have wells wherein you dig deep into the ground in search of water, and once you get the water, you will take a pump and then you will pump it out. Then you have canals which are there, right? So in the case of canals, what happens is that we receive, so in canals we receive water from reservoirs. So reservoirs are nothing but big places where we store water, or they take it from rivers. So what will happen is, this is a reservoir, no? They'll do a detour, okay? They will do a detour and they will build a canal so that the water can come there. So that it is divided into a main canal and a branch canal. So if this is a main canal, they'll do branch canal so that they get the water. Then you have river lift systems, which is something where from a canal, if you don't get enough water, or if it is insufficient or irregular, we see that a river lift system is something where water is directly taken from the river, right? And last but not the least, we have tanks, which are small storage reservoirs. So with this, we've looked at irrigation. Then we have cropping patterns. Now in cropping patterns, you have three which you need to remember. I am directly going to go to this part, right? You have mixed cropping, you have intercropping, and you have crop rotation. First thing you need to remember is the definition, right? So first you need to know definition. In mixed cropping, what happens? You have two or more crops which are grown simultaneously on the same piece of land, right? So in the same piece of land, two or more crops are grown. Now, how is mixed cropping different from intercropping? In intercropping, 2 or more crops are grown simultaneously, but there is a definite pattern, right? So in mixed cropping, I can grow it, maybe here I decided to grow some, or here I decided to grow some, or I divide half and half and I grow. But in intercropping, what happens is that, let me just make some space. So in intercropping, what will I do? I will take a piece of land and I will grow it alternatively. So here if I want to grow ginger, here I will grow cabbage, then ginger, then cabbage. So there is a definite pattern, and this is a key pointer they will ask you in your exams, right? So this is going to be happening. Okay. Now, of course, last we have crop rotation, where 2 or more crops are grown in the same area in a sequence of growing seasons, right? So here we have, if one crop is grown, so normally when we look at crop rotation, it is alternating, right? So they will be alternating one after the other. And second time we will alternate it with a leguminous plant. So what are leguminous plants? Leguminous plants are those plants wherein in the root nodules you have rhizobium that fix nitrogen. So crop rotation is done in such a way with the leguminous plants so that it can improve the fertility of the soil. So with this, we've looked at crop production management. Last one, we have protection. What do we need to protect our crops from? From weeds and from pests. So weeds are undesirable plants that grow along with our crops and that compete for your resources, while pests are undesirable insects that will compete or that will eat it up. So normally we end up spraying insecticides and pesticides, but see, insecticides and pesticides are chemicals, right? So they take a longer duration to, sort of, they have a negative impact, which is why what do? We also do weed control, right? By using mechanical removal, or we make sure that we take some precautionary steps such as proper seed bed, timely sowing of crops, intercropping, crop rotation. So we do it based on our cropping patterns as well, right? Then of course we have storage, where storage of grains are either in jute bags or in metallic bins, right? And of course that there are a lot of storage challenges as well, because even after you've made sure you've picked the right kind of seeds, you have practiced proper management techniques, you have made sure that you've protected your crops and you've harvested them also. But even after that, if you are not taking care, pests can attack, microorganisms can grow, moisture can benefit or promote the growth of microbes, which is why you need to always sun dry your seeds. You need to add neem, which has antibacterial properties, right? Or do large scale storage in silos and in granaries, right? Now moving on to the very last part before Menti, right? That is on this last portion. Yes, the last portion that is there, which is on animal based food items. Are we clear, everybody? Are we clear so far? Okay, very quickly, can you give me a quick thumbs up on the chat? Are we clear with whatever we have discussed? Yes? All right, very good. So, like I told you, improvement is something which we did very recently, a few weeks ago. So, it's very fresh in your memory. So, of course, I will. Ma'am, is this chapter important on your examination front? Very important, right? So we'll look at animal husbandry, okay? So animal husbandry is nothing but the practice. It is a scientific management of animal or rearing animals at a large scale, right? So what are we going to do? We are going to do a certain set of steps that we will repeat for animal husbandry. So see, when you are learning animal husbandry, make a table, keep these things in mind. You need to know about the varieties that are there. So you need to know examples of exotic varieties and indigenous varieties. You need to know how we will take care of them, right? How are their shelter, and what are the breeds, and what kind of food will we feed. You make a table out of it, make the pointers, three pointers. You will be well and good, okay? So now in animal husbandry, of course, course, we have cattle farming, poultry, fishery, and beekeeping. In cattle farming, of course, we mainly learnt about how cattle are reared at a large scale, because from cattle we get milk, right? And apart from milk, we also know that there are some animals which are reared for the fact that they do some mechanical labor. So animals from which whom we get milk, we call them as milch animals, while from whom we are going to do mechanical labor, we call them as draught animals, right? So in the case of cattle, we know that there are some indigenous varieties of cattle, mainly cows, right? So when we talk about milch animals, right, who give us milk, because we know milk ultimately is a rich source of protein and it's a wholesome food. Your milk givers include your buffaloes, it includes your cows, and to an extent goat, okay? But because in India largely we consume cow milk because it's kind of easier to digest by our body, so we tend to consume cow milk at a larger scale. Now, some indigenous varieties of cows include Gir, Khilari, Sindhi, and Devni. But what happened was, although indigenous varieties, right, your advantage of indigenous variety is that they were adapted to Indian climatic condition. Disadvantage, amount of milk that was produced was relatively less, which is why they went for exotic breed, because they were giving more milk. But what happened? What was the disadvantage? They were not able to adapt to Indian climatic conditions, which is where they came up with a hybrid variety or a crossbreed, right? So they came up with a crossbreed system where they crossed a exotic variety with an Indian variety so that it had the ability of both. So one example being Girlando. Now, of course, when we talk about the diet, we know that they will mainly have roughage or increased fiber, and along with that they will have concentrates such as gram husk and oilseed cake that will give them more proteins, right? And when we talk about shelter, we know that in case of shelter, they should be in well ventilated sheds. We must protect them from rain, heat and cold, and we need to make sure that we have a hygienic environment for them, because cows, if they are infected or they are not well, it will affect, right? It will affect the production of milk, which is why we need to make sure that they are regularly vaccinated from time to time. So this was all about cattle farming. Now moving on to poultry. Poultry farming is when we raise domestic fowl for what? For getting eggs and meat. Not chicken meat, just meat, okay? So what are examples of poultry? We have chicken, geese, turkey and ducks, right? Now, of course, in poultry, some of the desirable traits are making sure that, you have low maintenance, you make sure that you have good quality meat which is being produced, you have good quality eggs, and of course that you don't spend a lot of money taking care of them, but you get good produce at the end of it, right? So in the case, we know that broadly poultry are divided into layers and broilers, right? So layers are those birds which are reared for their eggs, yes? So you have leghorn and minorca, which are examples. Then you have broilers wherein they are reared for their meat. So here you have cochin and asil. So here again you have indigenous varieties and you have exotic varieties. These are some examples. Remember one example, because I know you cannot remember too many examples also, right? So it's very, very important. So in this case, you have indigenous variety, which is asil, exotic variety, which is leghorn, and of course a hybrid variety is what we prefer, that is leghorn and asil hybrid. And here again, we always prefer that the food that we eat, right, or the food that they eat is having good amount of, concentrates. So in the case of layers, like who are reared for their eggs, we make sure that they have rich sources of vitamin A, vitamin K and calcium in it so that the shells are sturdy. And for the broilers, we give them good amount of protein diet so that the meat is good, right? And at the end of the day, meat is what we consume them for. Then of course protecting them, vaccinating them regularly, and that's about it. Then of course you have fisheries wherein you have rearing of aquatic organisms like crabs, fish and squids and mollusks. So we call this as aquaculture. Now aquaculture technique wise can be of two types. You have capture and culture fisheries. Capture is when you capture them with a net, or you use some satellites or sonar to detect them and capture them with a net. Culture is when you culture it artificially, right? You make a pond or a tank and and then in that you will culture them. So here you are stealing them or you're taking away from their natural habitat, but in this case if you see, you are creating an environment, you are culturing them at a large scale, but you are not disturbing their natural habitat, right? Then of course culture fishery can be done in marine waters or they can be done in fresh waters. So here of course these are some examples that you can see. Now of course for marine fisheries or large scale production of marine organisms is what we call as mariculture, right? Again focus on the fact that I said marine organisms. I am not saying marine fishes. They are marine organisms, right? So pomfret, mackerel, tuna are all examples, and mullets, bhetki, pearl spots have high economic value. Then of course this is how we do capture fishery in marine fishes, right? And of course in inland fisheries you tend to do it in freshwater resources like maybe rivers, lakes, ponds and estuaries, right? Then here of course we know that composite fish farming takes place. Yes, so composite fish farming is when you have a tank and at 3 levels, you grow different species of fishes in such a way that they don't compete with one another for food. So you have bottom feeders, you have middle feeders and you have top feeders. So your top feeders mainly include like your catla, middle feeders include rohu, this will include karpa. And you put them in a tank and you rear them in such a way that it will not compete with each other. So this is what we mean by composite fish farming, where in a single tank with minimal resources you are growing maximum number of species. Last but not the least, right, for all of you who are like, whoa ma'am, it is so tiring. No, I am sure all of you, the last part, yes, very quickly, yay, in the chat, right? That is beekeeping or apiculture. So apiculture is the large scale production of bees, right, wherein we know that bees are cultivated for the honey that we get. We cultivate them in these apiaries that are there so that we get honey production. So some of them are Apis indica, Apis dorsata, Apis mellifera, and of course we know that these are the 2 popular ones. So with this we come to the end of today's session, right? Okay, ma'am, can you explain varieties once more? Are you asking crop varieties? Is that what you are asking me, Arjun? Are you asking me for crop varieties? Is that what you are asking? Okay. All right. Okay, very good everybody. All right, okay. So let's all now focus on the Menti quiz, right? The code is there on your screen. It's 1599293, okay? So let me just exit this. All right, are you all ready? Okay, you have all joined. Let me just quickly refresh the Mentimeter. It is loading. All right, everyone, you can quickly tell your friends also to join. I'm sure some of them would have fallen asleep also, but it's okay. Yes, all right. Please tell your friends and we can get started. Yes? I know today's session went a little long. Okay, everybody, so it is showing 6 to 8. All right, just give me one moment, okay? Just give me one moment. Yes, so I'm just going to put it on top of the screen. I'm going to load it once again, okay? Harish, can we just freeze the screen once? Can we quickly freeze the screen? I'll just reload it from my mail. Okay, everybody, just give me a moment. ma'am, are you going to sleep in office? No, no, I am not sleeping in office. Okay, they have just paused my screen, okay. Are you all able to hear me? Okay, everyone, they've just frozen my screen because we're trying to get this sorted, right? Yes, don't worry. I know you're not able to see because it's not loading. So just give me one moment. Yes, all right, Harish, we can get the screen back. Okay, can we get the screen back? Yes, all right. Can we have our screen back to normal, Harish? I think the students can see. I know the logo says 6 to 8. Just ignore that, okay? I am there. I am there. I am there. They'll bring me back. I had to fix this. No, I'm all alone here, so I was fixing it. So very quickly ignore the 6 to 8 that is there. Yes, but quickly join the code 1599293. Yes. Let me do full screen. Okay, let me just go back. All right. All right, everybody, join the code. Yes, I am visible. Code is 1599293. I can see 40 of you have joined. All of you are the ones who pestered me today, ma'am. Menti. Now, finally, we have Menti. Little late in the night, but it's okay. Yes? Answer bata dena Menti mein, please. Ha. Okay, all right. So now I'm back. Now I'm full free. You know, I've been with you all for 3.5 hours almost. So I'm going to quickly get started. I have 4378 in the live. So very quickly, everybody, get to Menti and we will join. Okay. All right. Very quickly, www.menti.com and go to 1599293. Yes. मैम मजा आ गया, आप सबको मजा आ गया। आज का क्लास तो बहुत ज्यादा लॉन्ग चल गया अभी तो। So I hope you all enjoyed and I hope you all focused in improvement in food resources. Yes? Okay, super duper excited. That's all I want, right? So very quickly, everybody. Yes, of course, we have ANTHE. We know about it. Let me go. Yeah, so we have 12 questions. We are going to be super fast, okay? Ma'am, diagram wala session cancel क्यों कर दिया? अभी तो Monday को अभी we will have the session. So don't worry, okay? And no, I will not teach till 12 o'clock. I want my students to sleep also. I don't want you all to be awake. No, you should take rest. Question number 1 on your screens. Yes? All right. How? Answer fast to get some more points. Okay, let's have a look. Which of the following plant tissue does not contain nucleus? Parenchyma, chlorenchyma, sclerenchyma, arenchyma. Very easy. Yes? Very easy question. Okay, all right, very good. We have, I think, 40 seconds, 45 seconds, but very quickly. Ma'am, it's showing second. Oh, I think there was a glitch there. Don't worry, it's fine. It's fine, okay. Parenchyma, chlorenchyma, sclerenchyma, arenchyma, which does not contain nucleus. Think logically, guys. Think logically. Next time I'll give you time, okay? Very good, everybody. Very good. So when we're talking about a tissue that does not have nucleus, we're talking about a tissue that is dead, right? Now your chlorenchyma is a modified version of parenchyma, which has your chloroplast in it. Arenchyma has your air sacs in it, right? So the correct answer is sclerenchyma. Yes. Okay. Don't worry, Amaino. Don't worry. Yes. Okay. Now let's move on to the next question. Okay. Let's have a look. Oh wait, I move fast. Okay, question number 3. Write your names very quickly. All right, I'm giving you 10 seconds so that you don't tell me, ma'am, you didn't give me time to do. Okay, everybody, are we ready? Yes? Are we ready? How to join Menti? How to join Menti? Go to www.menti.com, add the code. Everybody, please help your friends to join Menti. Yes? So this is question number 3 on your screens. Yes? Or I think you missed the first question. I'm so sorry. I think I jumped the gun there, but it's okay. The cell organelle discovered by Robert Brown contains a rod-shaped structure called as A, which is composed of B and C. What is A, B and C here? It's a little tricky, but an easy one, okay? Robert Brown discovered something which has rod-shaped structures called A. Rod-shaped, focus on the term rod-shaped, yes? Easy peasy, ma'am. Second question. I know. That was the second one. Yes. Let's have a look. Guys, what happened? Kya hua abhi? Robert Brown discovered, so the cell organelle discovered by Robert Brown is what? It is nothing but your nucleus. It contains a rod-shaped structure called as what? It consists of a rod-shaped structure known as chromosomes, right? Which are composed of what and what? Your chromosomes at the end of the day are made up of DNA and proteins that are packaged together to form chromatin material, right? So read the question thoroughly. So A is chromosome, B is DNA, C is protein. Yes? So are we clear? All right. Now moving on to question number 4 on your screens. Yes? ये कैसे हुआ? क्योंकि they are not asking what Robert Brown discovered. They are saying cell organelle that Robert Brown discovered has a rod-shaped structure A. That is why the correct answer is this. Focus on the question. But I also know you must be a little tired, but it's okay. Main thing is to learn, right? Let's have a look at question number 4. All right, on your screens. Select the incorrect statement regarding the depicted process. Okay. So here we have a process on your screen. 1, it involves 2 consecutive divisions. It occurs in body cells. It helps in growth and repair of tissues and organisms. In this process, daughter cells have the same number of chromosomes as the mother cells. This is a very easy one. Ma'am, it's little tricky. No problem. For those of you who are saying, ma'am, tricky, I will help you out. Very easy one, everyone. Very easy. All right. Okay. All right. Some of you got confused. Ma'am, Menti site crash ho gaya. No problem. Just reload the Menti site. Don't worry. Now you're like, abhi itna der wait kar karke Menti bhi crash ho gaya. Don't worry, okay, just reload it. So those of you who are confused with this particular question, right? See, it showed that there was a parent cell, right? And it gave rise to 2 daughter cells, no? Now look at the number of chromosomes that were there. There were 46 chromosomes in the parent, 46 in the daughter cell. Now, you have same number of chromosomes, that means it is equational division or mitosis, right? So in mitosis, where all do we find? We find it in body cells because I told you meiosis we find in sex cells or reproductive cells. Now mitosis is helping you in growth and repair, right? The number that is there will increase when you have increase in number of cells, it will increase in growth. In this process, daughter cells have same number of mother cells. Yes, it is there in the diagram. It involves 2 consecutive divisions, which means that after one daughter cell is formed, that is one division, one more division should happen. That is not something we see in mitosis. We see that in meiosis. So are we clear? Yes? Are we clear? Okay. Doing tukka. Who is doing tukka? I think 7th graders are here. I know I skipped first question. Very sorry, guys. We'll go back to this. Can you explain once again? See, you had one daughter cell, one mother cell, right? Yeah. So as you know, you had one parent cell which had 46 chromosomes, giving rise to two daughter cells with 46 and 46. So that is your mitosis. So you've understood that, you've come to the fact that it's mitosis division. Now mitotic division occurs in body cells, helps with growth and repair, and because the parent cell and the daughter cell have same number of chromosomes, option D is also correct. They are asking select the statement which is incorrect, right? Okay. Question should have been select which is correct. Which is why this is the correct. This is incorrect about it, right? This is incorrect about what was told. It is telling it involves two consecutive division. It doesn't. Mitosis doesn't have it. Meiosis has it. Okay? Yes. All right. Abhi kya hua chat pe? Lot of you are saying kya. Next question. Okay. Abhi all of you all focus. Okay? All right. Question number 5, everyone, on your screens. Okay. Ma'am, I want to appreciate you knowing the answer. Did I reveal the next one? Did I do that? Okay, on your screens. Yes? All right. On your screens, and let's start. The solution is said to be hypertonic solution if the solution has a lower water concentration than the cell, the solution has same water concentration, solution has higher water concentration, the solution has a lower solute concentration. This is like a tongue twister. I spent a lot of time in this, okay? So you all should be able to answer this. Hyper means more. I'm giving you a clue. All right. Very good, everybody. Very good, but I can see lot of you got confused. Okay, see, we're talking about what is hypertonic solution. Hypertonic solution will have more solute, less solvent. So if it is having more solute, then water concentration will be lower, right? That is why in hypertonic solution, what is happening? Water is moving outside the cell. In this case, where some of you would have gotten confused, solution has higher water concentration, right? That is not the case, no? Which is why this is the correct answer. So are we clear now? Yes. Okay, very good, everybody, very good. Now moving on to the next one. Question will come, leaderboard will come after this particular question. Yes? Okay, on your screens. I can see people are still joining. Ma'am, why A? A because lower concentration of water. Hypertonic is when hyper is more, right? More amount of solute. That is what you need to remember. More solute, less water. All right, let's have a look. Which of the following are exotic breeds? Braun, Jersey, Brown Swiss, Jersey Swiss. This is an easy one. This is a very easy one. But improvement, I hope you all paid attention, okay? All right. Very good. This is a very simple one, guys. Time's almost up. This is why you should pay attention when I did improvement, right? Some of you were so confused. What is Braun? What is Jersey Swiss? Chuma, they made names. Team has only made their own this one. So, which of the following are exotic breeds? They are your Jersey and Brown Swiss. Okay? These are the correct answers. Yes? By the way, help is 1 and 3. Yeah, it's 2 and 3. 1 Braun, there's nothing known as Braun. Okay? That's why the correct answer is 2 and 3. So, very quickly, let's have a look at the mentee. Yes, we have the leaderboard, right? Quickly, let's have a yes, please don't spam on the chat, guys. Don't spam. So on top, of course, we have Himanshi, who is the fastest. We have Nishta, Soumya, AV, Pratik Sharma, Kalash, Kushagra, Einstein, Shritama, and Shifa. Yes. Ma'am, typing by mistake. No problem, Soham, no problem. I didn't join Menti. Go join Menti. We are having a live quiz, right? A lot of you have got 13th place. Very good, very good. So now let's move on to the last set of 6 questions. Let's wind this up quickly. Okay, let's get started. Answer fast to get some more points. Let me stand on the other side. Which of the following statements is true regarding the Haversian canal? I know I didn't go in detail with this. It contains a slender process known as osteocyte. It is a type of muscular tissue. It nourishes the neurons. It contains blood vessels and nerve fibers in the bone. Okay. It is true regarding Haversian canal. Okay, see. You think about it very logically. 2 options you will eliminate like that only. Then you have 2 more and you can try it out. Yes. This is going to be a simple one. See, Haversian canal, I'll give you I want to give you a clue, but it's okay. See, 2 you will eliminate, I know. Yes, Arab, hello. See, Haversian canal, everybody say, oh, canal means it will be something thin and narrow. So everybody is like, it contains a slender process of osteocyte. Osteocyte, we know, are nothing but bone cells, right? So bone cells, how will it consist of just one single process? That's not a canal, right? So that is not what a Haversian canal is. It is a canal which we find inside the bone that is having your blood vessels and nerve fibers, right? So of course this is a little beyond. So for your anthe, you might get questions like this. So think logically. It's saying consists of a slender process. So are we saying a canal is just one thin process? No, right? That will not be there. Nourishes neurons? No, no. Is it a type of muscular tissue? No, no. Right? Now moving on to the next one. Question number 8. Everybody is here? Yes, all right. Okay. On your screens, everyone. Match the following. Let's see what is our match, right? Okay, there is no image on screen. What do we do? Ignore this question. There is no image on screen. You can't do anything. You are not able to see any image, right? None of you are able to see image. Menti has given heart today. Menti is like, no, I'll not give you image. Right, so it's okay. Tukka maro. Let's see what is there. You can just randomly guess one thing and see. Just put something. I don't know what is there in this. All right, everybody, we are almost there. No photo, just give tukka. We'll see. We'll go on to the next question. For those of you who gave a guess, very good, well done. Menti is like, oh, it's already 10:30, time to sleep. Why are you quizzing? I mean, why are you playing quizzes on my site now? Okay, all right. Okay, let's have a look at the 9th question. On your screens, everyone. Chromosomes are composed of. After giving that previous question, you should answer this. RNA and lipids, proteins and DNA, RNA and DNA, or lipids and DNA, right? Sahi aaya ma'am, tukka lol. Yeah, you never know what will be there. Yes, done ma'am, easy question. It's an easy one. And guys, if you liked this session, and I know today's session was long, but if you liked it, do not forget to hit that like button and do not forget to subscribe to our channel, okay? Because see, we want to make sure that we prepare you well for all of it. Yes? So don't forget to like our video. Do not forget. And subscribe. ऑप्शन बी को लॉक किया जाए. ठीक है, हम देखते हैं कि क्या आंसर है. अरे वाह, correct answer is proteins and DNA, because we've just learned that the rod-shaped structures that are chromosomes are there. Yes, now moving on to the next one, right? Question number 10. 3 more questions and we are done, right? We are done. Ma'am, real pain is being... Yeah, I know. Let's have a look. Okay. Okay, all right. Function of 1 is to release energy in the form of adenosine triphosphate. Here 1 is what? Leucoplast, ribosome, mitochondria or Golgi apparatus. Super duper easy, right? Super duper easy. Ma'am, online exam kaisa dena hai ANTHE ke liye? From home only you can give the exam, right? So you should have like a laptop and everything ready with you. So you just need to give it from home. Arey Sahil, kya ho gaya? Oh no, don't worry, don't worry. It's okay. Don't worry, Sahil. Just refresh it and try again. You must be able to access it, okay? Akansh, I hope I've answered your question. ये क्या पूछ लिया? Very good. Correct answer is mitochondria. Now moving on to question number 11, which is the second last question. Let's have a look. Ma'am, can we use Google? Please don't use Google. After 3 hours of class, don't be Googling it. Okay. Which of the following plant tissue allows bending of various parts of the plant without breaking? Parenchyma, chlorenchyma, sclerenchyma or collenchyma? Yes. Oh, time's up so fast. Or did you all just answer that fast? Okay, very good everybody, very good. The correct answer is collenchyma, right? Because when we talk about bending of various parts without breaking, we are talking about flexibility, right? So here it is collenchyma. Again, I can see somebody is like, ma'am, it should have been B and I clicked A. It shouldn't have been A also because this is for storage. Parenchyma is for storage. Chlorenchyma is to mislead you with collenchyma. The correct answer is collenchyma, right? Now we'll move on to our last question for today. Yes, last question. Yes, all right everybody, last question. Okay then, let's have a look on screen. Answer fast to get some more points. Find out the wrong statement from the following. Okay, we have 4 statements. White revolution is meant for increase in milk production. Blue revolution is meant for increase in fish production. Increasing food production without compromising environmental quality is what we call as sustainable agriculture. None of the above. This is very easy, guys. Super easy. Super duper easy question. Yes? Ma'am, I see session at last. Oh, okay. It's in Feb Army Cake World. It's in Feb. See, 2, even if you don't know, you'll be able to answer this. That's how easy it is. Very good everybody, very good. We are talking about find out what is the wrong statement from the following. Increasing food production without compromising environmental quality is what we call as sustainable agriculture. Yes, sustainability is when you don't compromise the environmental quality. I told you, no. What happened was in the initial revolutions, we were compromising or overusing our natural resources, right? But we want to do something where we don't overly exhaust it, but we increase the production. That is what is sustainability, right? White revolution is for milk, blue revolution is for fish. So technically none of it is wrong. So trick question, yes? And of course, with this guys, we come to the end, right? So last leaderboard, let's have a look at the leaderboard. Okay, so let's see. Kalash is the topper. Congratulations, Kalash. We have Pratik, AV, Soumya, Ronald, Kushagra, Himanshi, Shritama, Archita, and Nishita. Very good, everybody. Very, very good. Yes. So with this, of course, I come to the end of today's class. Now I know today's class was long, so much to study. But trust me, man, I will not be able to pull this off without you guys having so much interest. Even though you were telling me, ma'am, I think I'm feeling sleepy. Ma'am, I don't know if I'll be able to pull it through. You did technically revise. And if you see so far, your physics and your chemistry revision for all your chapters has already been done. Right? And with this, if you see, we've learned all the important chapters that are there. Now, of course, very quickly, everybody, If you still have some doubts which are pending from this chapter or some of the chapters, maybe I missed your doubts during the session or you still do not have conceptual understanding, you let me know in the comment section below, okay? Because I will definitely reply to you, and if you have been somebody who's got my messages, you know that I will reply to you no matter what, right? And if you enjoyed this class, you should also tell me that you enjoyed the class, right? And of course, next for 9th grade, you have math session, which is on November 1st. Khushbu ma'am will be taking, and then you have chemistry. And with this, if you see, your science and math revision for all the chapters that we've covered so far is done and dusted. So do not forget to like this video, share it with your friends, subscribe to our channel, because we have got you covered. We will take care of you, prepare you, make you amazing students, right? So yes, thank you so much everyone. Thank you so much for being wonderful students. Go home, take rest, not go home. Take rest, sleep well, and have a nice day tomorrow. Yes? All right. Okay, everybody. So thank you so much for today. Hoping to see you very soon again. Bye-bye and have a nice day.